Good morning to everybody. I think we can begin this conference. So we have translation in French, in English, and in German. And so I will switch to French. Donc merci beaucoup à vous toutes et tous d'être là aujourd'hui. Merci aux différents intervenants. Quelques mots déjà de présentation, puisque certains m'ont déjà vu dans des conférences déjà sur ce sujet des nouveaux OGM ou sur le glyphosate. Donc Christophe Clergeau, je suis le shadow rapporteur pour le groupe socialiste sur le texte NGT pour les bois qui viennent. Et je vais être aussi le rapporteur de la commission Envie sur le texte sur les semences reproductive material. Et je pense qu'il y a d'ailleurs un lien entre ces deux textes et je crois que c'est un enjeu important de voir comment on travaille sur ce que sont les produits qui sont mis à la disposition des agriculteurs et les produits alimentaires proposés aux consommateurs. Alors merci à vous tous et toutes d'être là. Il y a de la concurrence ce matin puisqu'il y a aussi en commission Agri la présentation du texte NGT. Donc il y aura sûrement des gens qui vont faire des allers-retours entre les deux conférences. Et je pense qu'on est au moment où il faut véritablement ouvrir le débat et sortir des postures. C'est le moment parce que le calendrier est en train de s'accélérer. Nous avons ici au Parlement un débat sur le calendrier puisque la rapporteure veut accélérer les choses et nous propose une date limite d'amendement le 15 novembre avec un vote en envie en janvier et en plénière dès fin janvier. C'est une accélération assez subite, contraire au calendrier qu'on avait 15 jours plus tôt. Donc il y a un débat au sein des coordinateurs d'Envie pour qu'on ait un peu plus de temps pour faire ce travail sérieusement. Et pourquoi ? Parce qu'il faut sortir de positions de principes auxquelles certains veulent nous acculer. En gros, on veut faire en sorte que le débat ce soit pour ou contre la technologie. Et je ne sais pas s'il y a des parlementaires qui sont pour ou contre la technologie. Je connais des gens qui sont pour a priori. Je n'ai pas encore rencontré de députés qui sont contre a priori. Et moi, le débat que je veux poser, et c'est pour ça que nous avons organisé cette matinée, ce n'est pas d'être pour ou contre la technologie, mais c'est de se poser la question de savoir comment une société utilise une technologie, une innovation, pour la transformer en produit proposé aux agriculteurs, proposé aux consommateurs. Et pour avoir ce débat-là, si on se réfère à tous les débats que l'Union européenne a eus depuis 40 ans autour de la mise en place du marché intérieur, on a toujours fait deux choses. Regarder la science, toute la science, et se poser la question de savoir comment on prend en compte la science. Et je remercie l'EFSA d'être aussi avec nous ce matin, parce que ce n'est pas facile de déterminer comment on prend en compte la science, surtout dans des domaines où il y a de la science émergente, et on va en avoir aussi des présentations. Et donc comment on utilise la science pour fonder la décision publique, et comment on prend en compte les droits des citoyens, qu'ils soient agriculteurs, qu'ils soient consommateurs. Nous héritons d'une tradition de dizaines d'années où un haut niveau de protection de la santé et de l'environnement, le libre choix des consommateurs et des producteurs, des fermiers, sont des acquis, pour certains des traités, pour d'autres de la législation. Moi, je pense que ce sont des éléments fondamentaux des traités qui doivent aussi être pris en compte dans la manière de construire une législation. Donc je ne vais pas moi-même ouvrir tous les débats, ils vont être éclairés au fil de la matinée. On a essayé de ne pas avoir trop d'intervenants pour bien laisser le temps à l'EFSA puis à la commission de répondre et d'être dans la discussion. Mon collègue Martin Osling des Verts va nous rejoindre pour pouvoir réagir aux présentations scientifiques et je compte sur un échange avec la salle. Et cet après-midi, j'organise aussi des réunions avec les parties prenantes pour prolonger les discussions de ce matin et pouvoir avoir un contact direct pour entendre les arguments des uns et des autres. Et pour terminer, je dirais une chose très pratique, parce que certains seront peut-être repartis pour mes conclusions en fin de matinée, c'est si vous avez des amendements, envoyez vos amendements. Maintenant, puisque comme vous le voyez, nous avons des délais qui sont brefs. Donc avec mon équipe et avec les uns et les autres, nous sommes déjà dans le travail de rédaction des amendements et de préparation pour faire face aux échéances législatives. Donc nous sommes à l'écoute de toutes les propositions qui vont venir, parce que par rapport au texte initial de la Commission, si on veut réellement prendre en compte la science dans le processus de décision, si on veut réellement prendre en compte les droits citoyens, il y a beaucoup de points qui doivent faire l'objet de débats et qui peuvent faire l'objet d'amendements. Merci à Martine d'être avec nous. I told everybody you are coming. Et maintenant, moi, je vais aller me mettre au premier rang pour bien écouter et je vais laisser Astrid piloter ce premier panel en remerciant donc nos amis de Test Biotech qui apportent beaucoup dans cette réflexion comme organisation scientifique en lien avec de nombreux chercheurs en Europe. Donc je laisse Astrid piloter ce premier panel et puis je reviendrai piloter le second. Merci à toutes et à tous.
Thank you very much, Mr. Clergeau, for this inspiring opening statements. And welcome also from my side to everybody joining us here in the room and also online. My name is Astrid Österreicher and I'm an EU policy advisor for Test Biotech. And I have the honor today to be chairing our first panel on the sanitary and environmental risks related to new GMO plants. The question is, what do we know? We're dealing here with a highly complex and controversial topic where simple slogans such as "listen to the science" do not work anymore. I think we all agree, policymaking should be science-based. But you cannot just listen to the science, because the scientific community is deeply divided on the risks and potentials of this technology. So I thank also Mr. Clergeau for pointing out that we have to take into account all of the science here and not just listen to a majority opinion. It is encouraging to see that even though you are all racing against the clock with these very tight deadlines, you are taking the time today to take a profound look at scientific questions underlying this debate, to inform your political work. We are going to start the scientific presentations with Franziska Koller today. She holds a PhD in microbiology, and she's currently working for the project Genetic Engineering and the Environment, which is a scientific research project funded by the German Federal Agency for Nature Conservation. She will fill us in on unintended changes caused by the CRISPR-Cas gene scissors. Now, this is quite interesting because I'm sure that all of you who are working at the Parliament are being bombarded right now with email and other kind of messages, according to which we're dealing here with a very precise technology that only leads to intended changes. Well, now we will hear from Franziska why this is only half of the truth, and that there can also be unintended changes besides the intended ones. Franziska, please go ahead.
Thank you.
So we're waiting for the slides, I think.
Yeah, thank you. Good morning. Thank you for the opportunity to present our work here today. I will talk about unintended genetic changes and effects in NGT plants that require risk assessment. I will split my presentation into two parts. Firstly, I will talk about unintended genetic changes, and here I will summarize the result of our recently published review. In the second part, I will talk about unintended effects on the plant and the environment, and here I will present an example we are currently examining. Oh, sorry. The publication I just mentioned, we review findings in regard to the unintended genetic changes that can be caused by the applications of NGTs. While some of the types of genetic changes might also be observed in conventional breeding, there may be some differences in regard to the probability that these changes occurring at specific sites in the genome. We categorized these unintended genetic changes specific for NGTs and came up with 5 categories to distinguish them. The first category comprises unintended genetic changes resulting from the insertion of transgenes using old or established genomic techniques. Currently, we see that in almost all NGT plants, there are old genomic techniques used. And by using these techniques, the integration of the DNA of the gene scissor is random. This results in a transgenic plant, which may show unintended genetic changes, which can lead, for example, to the disruption of genes and also affect epigenetic regulation. In order to remove transgenes, researchers are using segregation breeding, but sometimes the transgenes or parts of it remain in the genome unnoticed. The second category comprises unintended insertion of transgenes, because during the NGT application, there's often transgenic DNA in the plant cell, for example, DNA from bacteria like Agrobacterium, which is used to transfer the DNA of the gene scissor into the cell. And there are examples where parts of this bacterial DNA are integrated unintendedly into the genome. The third category comprises unintended on-target and off-target changes. On the one hand, there are various examples describing the off-target cleavage, for example, DNA which has similarity to the target region. On the other hand, there are also unintended genetic changes at the on-target site. For example, large insertions or large structural changes. And it was shown that these are not always easy to detect. Due to the technical potentials of NGTs, these unintended genetic changes might occur at genomic sites that differ compared to conventional breeding. The next category is about chromotripsis-like effects. Chromotripsis is a genomic event where possibly hundreds of chromosomal rearrangement can happen in a single catastrophic event. This process can result in large rearrangement and loss of DNA. In humans, it is described that this chromotripsis is associated with cancer, for example. Publications showed that this can happen at double-strand breaks introduced by NGTs, and further that the use of NGTs can increase the likelihood of chromotripsis to occur in mammalian cells. Recently, it was shown that chromotripsis can also occur in plants, and therefore the use of NGTs in plants may impact the likelihood of chromotripsis to occur at specific sites in the genome, especially in multiplexing approaches. The last category comprises of unintended genetic changes that may cause the formation of new gene products. The genetic changes by NGTs can result in various outcomes regarding the formation of gene products. For example, it is possible that no more mRNA is built, but it's also possible that new mRNA can be unintentionally formed, which can then lead to the production of shortened or new proteins. These can still carry out function or have altered functions in the cell. To sum this up, unintended genetic changes by NGTs can affect small and large sections of the chromosomes and result in unintended gene products. The site of mutation, their genomic context, and their frequency in regard to specific sites can differ when compared to conventionally bred plants. The biological effects can therefore be different and may cause specific risks for health and the environment. These findings should be considered in the molecular risk characterization and risk assessment. To comprehensively identify and assess the unintended genetic changes specific for NGTs, a combination of multiple approaches is important. In addition, risk assessment should include omic methods to identify associated unintended biological effects. Sorry. I will now come to the second part of my talk about unintended effects on the plant and the environment. We analyzed current NGT applications in the plant family Brassicacea and had a closer look on the oilseed crops, oilseed rape, pennycress, and camelina. We chose this plant family as an example as Brassicacea. It is known that the genome of Brassicacea is highly complex. Therefore, conventional breeding methods are described to have restrictions and shortcomings. Using NGTs, genotypes and phenotypes can be obtained that would be hardly achievable using conventional breeding. We counted more than 50 NGT applications in these oilseed crops, and almost all of them would fall into the category 1 of the Commission proposal. Looking at the intended trait, most of the applications aim to change the oil content or the fatty acid composition. On the other side, many applications would have or have positive effects on the plant fitness, like prolonged flowering or more seeds or superior growth. Looking at the effect that these applications can have, we identified some unintended effects and risks for the plant and the environment. A lot of NGT applications aim to change the fatty acid composition, in particular to reduce polyunsaturated fatty acids in NGT plants. But the decrease in these fatty acids can negatively affect the health of pollinators, as pollinators may suffer a deficiency in these polyunsaturated fatty acids in NGT monocultures. And for honeybees, for example, it was shown that a deficiency in these fatty acids greatly impairs honeybees' brain function. In addition, Brassicacea are native to Europe, and a lot of Brassicacea species can hybridize with each other. In addition, many Brassicacea have weedy characteristics, a long seed dormancy, and pollination is possible over long distance. Therefore, under European conditions, the cultivation of NGT oilseed Brassicacea may not be easily controlled. There are already documented cases where transgenic oilseed rape spread and crossed out into weedy species. The increasing fitness we see in a lot of NGT application can further increase the risk of spread and persistence. The release of NGT Brassicacea may result in spontaneous crossing and therefore in uncontrolled spread of NGT traits and combination of different NGT traits. As most of these NGT applications would fall into the category 1, their further stacking and crossing is possible without further restriction and monitoring. But with increasing complexity and intended or spontaneous combination of these traits, undesirable pleiotropic effects, also side effects, and unintended consequences become more and more difficult to predict. I want to shortly summarize these findings. NGT can cause different intended and unintended genetic changes and effects compared to conventional breeding. If unintended genetic changes and unintended effects are overlooked, they may endanger health, environment, and agricultural production. Therefore, it is mandatory that unintended changes and unintended effects have been taken into account in the risk assessment of NGT plants. Thank you for your attention.
Thank you very much, Franziska. I think it was very clear presentation, but I don't know if it was clear for you also. I want to give the chance, if you have a question just on the understanding, to raise it now. If you have questions on the content, that will be part of the discussion later. But if you have a question just on the understanding, I can take it. But I think it's fine. Everybody understood. I see nobody raising their hand, so it's fine. And I think you made some very important points. You know, if we overlook these unintended changes, then this can really lead to damage for environment and health as well. Just to give you a little bit of political context also, these unintended changes are for the moment not taken into account by the Commission proposal. The Commission proposal only takes into account the intended changes. Recently, the European Council asked for a scientific justification of the Commission proposal, but again, in this justification, which was published last week, the unintended changes are not taken into account. So I think this is one of those questions which are still open for discussion, how they should be taken into account. But I think if we take the precautionary principle serious, we cannot just overlook them. We will now move to the second presentation, which will be given by Mrs. Sarah Agapito. She's sitting here. She completed her PhD in microbiology. She completed her PhD in plant genetics, and she's currently working as a research professor at the Norwegian Independent Research Institute, NORS, which aims to facilitate informed and sustainable choices for the future. So just what we need. Sarah has participated in international research projects on both identification of NGTs as well as their risks. The main goal of her current research is to uncover the roles of transgenes in gene regulation in plant systems and to apply that knowledge to assess their safe use in food and the environment. Sarah, we're excited to have you today and to listen to your presentation.
Thank you again for this opportunity. I'm sure I'm going to learn too with this exchange. Okay, let's see. Yeah, so a lot of the points I'm going to raise in this talk refers to this specific paper we published in January. So this was before the proposal was published. But the reason is because what we define as null segregants, it's quite similar to what the Commission is proposing to define as NGT category 1. So I will not bombard you with a lot of scientific references, but they are there in this paper. It's open access. Many of you have seen the text. I just would like to recall that the definition of NGT category 1, and that's mainly what I will discuss in my talk. So the Commission states that given that these NGT category 1 are equivalent to conventional breeding techniques, then the risks are comparable. So it assumes that equivalence correlates with the level of risk, and that's a challenge that I'm going to take with this talk. And in order to establish equivalence, the Commission proposes these criteria in Annex 1. And as you can see, I'm not going to go through each of this, but it's a lot related to the number of nucleotides, DNA nucleotides that are changed in the NGT plant. So there are many questions related to this criteria. From a plant geneticist point of view, there are technical semantics, whether there is 1, 2, or 3, whether they are in target site 1, 2, or 3. To my understanding, there is a step back and a question on whether these techniques, in fact, they improve the speed and the scale that these products are going to be used, and that's not comparable with neither natural products or products from conventional breeding. So these thresholds, they are arbitrary. There is no correlation between 20 nucleotides and risks. And I think Francisca gave us very good examples of how unintended changes can be smaller than 20 nucleotides or even larger and have risks. And they are also loose because they are definitions that are not clear. So the process by which perhaps EFSA is going to develop further on, if the proposal is taken as it is, that will be very important to establish the risk profile. So the way that the proposal is written is that a risk category is decided then by a threshold-based trigger, which is the number of nucleotides changed. And these are normative judgments of equivalence, which are arbitrary estimates of how many nucleotides must change to create hazard. And the reason why I'm linking this to risk is because the Commission is proposing to take equivalence as correlation to risk. This kind of discretion would remove significant activities of gene technology beyond routine public accountability. And one example is the application of CRISPR in the environment. So this has been spotted by the ad hoc technical expert group in the Convention on Biological Diversity, which holds the Cartagena Protocol, the International Treaty on GMOs. They have highlighted this possibility. And we have seen this, for example, already being applied in Brazil. So you can modify plants in the field, not in the lab anymore. And these products would fit into NGT Category 1 of the proposal. But what is risk? Because we are so far away from discussing risks from the Commission text. Risk is not only the NGT product, the NGT plant. Risk is where the environment is going to be, where the NGT is going to be exposed in the environment. What type of environment is that? It's about protection goals, and it's about how these plants grow and the impact they have in the environment. So it's a probability of harm and also severity of harm. There are three key steps in risk assessment. One is identification of hazard. The second is assessment of exposure. And the third is proposed mitigation strategies. Because we are now working with zero risk, there is also risk, but we can, through regulation, decide on mitigation strategies. There's plenty of examples on how to conduct risk assessment. There's EFSA with their guidelines. There is also the guidance on risk assessment from the Cartagena Protocol. And we can extract this information from these guidelines. I would be happy to have a session just to discuss on what are the methodologies that we could use for NGT on risk assessment. But the proposal is not about risk assessment. The proposal is about deregulating them from risk assessment. So this question remains, how many nucleotides must be changed to create a hazard? We don't know this answer, and this answer cannot be just a magical number of 20 nucleotides. I've listed a few characteristics of NGTs for us to discuss and debate. NGTs are a product of gene technology. And I challenge you to go to the EU SAGE database, which lists more than 800 papers that have modified genetically plants. And pick 10 papers and check how they have modified these plants. You'll be surprised that 9 or even 10 have used classical genetic engineering techniques to modify these plants. So NGT Category 1 are in fact an end product of cumulative interventions, not only CRISPR interventions, gene editing interventions. At the same time that they increase efficiency and the target site, they also increase efficiency at off-target sites to modify these locations, as well presented by Francisca. So CRISPR, which is the most used technique, does not work like a lock and a key. It has a range of binding affinity to the genome, and that is what leads to unintended effects in other genomic locations. And most importantly, these secondary targets are largely beyond our ability to comprehensively identify them in advance, because they are case by case. They will depend on the cell environment. They will depend on the genomic composition. Therefore, verification is needed. It cannot be assumed a priori there won't be these modifications in plants. So to our understanding, from the risk assessment perspective, any sincere effort to demonstrate that NGT Category 1 was equivalent to a conventional plant would be as demanding, as necessary, as assessing their risk under the current GMO regulations. But I would like to finalize my talk with the different dimensions in risk assessment, dimensions that range from biological to socioeconomics. But of course, in my competence, I can only speak about the biological dimension. But I think there should be also other dimensions that are important for the impact in society, for example, socioeconomics and ethics. So we conclude, the authors of the paper conclude that gene technology, even when used to make what we call no segregants or NGT category 1, have characteristics that make regulation, risk assessment-based regulation, a reasonable option to mitigate potential harm. Thank you.
Thank you very much, Sarah, for this very clear presentation. I think it was very clear, but again, I want to give the chance if you have a question for our understanding. Otherwise, thank you again, and I think as Test Biotech, we came to the same conclusion that actually these criteria are arbitrary and do not allow to determine the equivalence. But the question now, of course, is what needs to be done that this equivalence can be established. But I think that will be a question that we can discuss during the final discussion. Franziska, you have a question?
Franziska Achterberg from the Greens IFA group. Sarah, I was wondering, you used the term classical genetic engineering techniques or something that are being used. Earlier we heard about insertion of transgenes. Can you explain what you mean by classical GMO technology or GMO techniques?
Yes, I'm sorry, I didn't list that. So what we call classical genetic engineering techniques are the techniques used to make the GMOs from the previous, from Directive 2001. So this is biolistics, random integration, bombardment of the genome, agrobacterium, which infects the cell plant and transfers transgenes, electroporation, so electric shocks to get transgene inserted in the cell also randomly. What other? Yeah. And the reason is because one needs to transfer the CRISPR machinery to the inside the cell of the plant. And there is only one way to do it without using this classical genetic engineering techniques, which is transferring the protein complex. We do this in our lab. So we develop a lot of model plants that are gene edited so that we can study effects. But the efficiency of doing this is very, very low in plant cells. So it's less than 15%. And even that, we need to make the membrane of the cell permeable. So we use either heavy chemicals or electroporation. But that's not used in these papers because it's not efficient. Most of the papers will still transfer plasmids into the cell. Of course, just a final note, they are going to back cross with a conventional plant to extract the transgene that has been inserted. But that's not a straightforward process, and that verification is needed, and it's missing in the proposal.
Thank you very much, Sarah, for this clarification. We're now going to move to our next speaker, who is going to join us online, Mr. Yves Berthaud. I'm going to switch to French to introduce him. Yves Berthaud est biologiste de formation et il est directeur des recherches INRA au Musée national d'histoire naturelle. Je pense qu'en France, il ne serait même pas nécessaire de le présenter car tout le monde le connaît. Si vous demandez à des ONG qui travaillent sur le sujet des nouveaux OGM en France, ils peuvent vous pointer vers un expert scientifique sur la question. Ils vont vous pointer vers Monsieur Yves Berthaud. Bref, on est très heureux de l'avoir pu recruter comme intervenant aujourd'hui et vous allez comprendre pourquoi. Yves, je te passe la parole.
Merci Astrid. Désolé de ne pas avoir pu vous rejoindre à Bruxelles, mais j'avais différentes obligations par ailleurs. Donc les NGT sont une de ces nouvelles séries de promesses d'un avenir radieux qui nous rappelle celle précédente sur les OGM transgéniques il y a 40 ans, le clonage il y a 25 ans. Je vais vous parler des NGT sur la prochaine diapositive comme des manipulations. Sur la prochaine diapositive. comme une technique de manipulation qui permet une réécriture du génome. Je dis bien réécriture du génome, car parler d'édition, ça voudrait dire que je connais la langue et la grammaire. Or, actuellement, nous en sommes au niveau de la connaissance des génomes et des épigénomes, au stade de réécrire des hiéroglyphes avant la découverte de la pierre de Rosette par Champollion et son décryptage. Sur la diapositive suivante, je vais vous rappeler, sur celle encore, la prochaine, je vais vous rappeler que le végétal est un ensemble de tissus non mobiles qui ont développé des capacités particulières d'adaptation à l'environnement. Les cellules et les tissus communiquent entre eux via des protéines, des hormones, de l'ADN, des ARN messagers. De même, les comportements cellulaires sont complexes, communiquent entre eux. Il est remarquable que l'évaluation des risques, comme celle que j'ai effectuée pendant un certain nombre d'années au Haut Conseil des biotechnologies, ne prennent en compte que le noyau, mais pas les autres compartiments contenant eux aussi des acides nucléiques, tous ces organites qui influent et sont influencés par les modifications effectuées au niveau du génome. La diapositive suivante vous rappelle que, en fait, les NGT ne sont qu'une des promesses technosolutionnistes pour nourrir le monde, assurer la sécurité alimentaire, lutter contre le climat, etc. En fait, c'est une des promesses classiques et qui repose, diapositive suivante, sur la croyance que sur plutôt l'idée, la reprise de l'idée de la biologie moléculaire des années 1970, celle des prix Nobel, Wolf, Monod et Jacob. L'idée de base des techniques NGT, c'est que le génome est un ensemble de briques interchangeables et que les sélectionneurs vont pouvoir, puisqu'ils sont toujours à la recherche d'une diversité qui a été supprimée, réduite fortement entre la domestication et la sélection variétale des dernières décennies, et que ces briques vont permettre de passer d'une espèce à une autre, voire entre variétés, quand il s'agit de modifier. Cette sélection variétale, en fait, basée sur des systèmes mono- ou oligogéniques, correspond à une course effrénée à la diversité. Or, diapositive suivante, elle se heurte à la complexité de ce que l'on a des génomes. Je vous rappelle ici juste celui du colza, du colza. L'approche réductionniste nous parlait il y a quelques années d'un génome de référence à partir duquel on pourrait échanger des briques entre espèces, voire entre variétés, alors qu'on sait maintenant que les génomes sont bien plus compliqués et qu'il faudra bien plus de travail et d'argent pour essayer d'en tirer quelque chose. Car les génomes varient au sein d'une même espèce, entre populations, mais aussi entre voisins pour les plantes qui ne sont pas des clones, avec des parties indispensables et des parties variables. C'est ainsi qu'on découvre que le génome du maïs en altitude est beaucoup plus petit que celui dans les plaines mexicaines. Dans la diapositive suivante, je vous rappelle que le génome n'est pas un système linéaire, mais un système tridimensionnel qui fait que la moindre variation à un endroit peut avoir des effets distaux, c'est-à-dire à distance. Une mutation ponctuelle, par exemple, peut modifier le positionnement dans un domaine lointain, ce qu'on appelle un TAD, et changer la régulation, l'expression d'une protéine qui n'est absolument pas à côté de la mutation que l'on a induite. Ceci pour revenir aussi à un autre problème déjà évoqué par exemple à propos de la taille des nucléotides. La taille de 20 nucléotides en deçà de laquelle une modification génétique intentionnelle ou non intentionnelle serait sans véritable conséquence et sans aucune base scientifique. C'est un peu comme dire qu'une voiture sur 3 roues peut rouler, au moins dans certaines circonstances. Il est actuellement nécessaire de séquencer des milliers de pangénomes pour se faire une idée de ces briques, et ce n'est probablement qu'un début, si vous vous rappelez qu'en ce qui concerne l'homme, ce sont des centaines de milliers de pangénomes qui sont en cours de séquençage. Quand on se rappelle, sur la diapositive suivante, les difficultés qui existent pour essayer d'établir une corrélation entre le génotype et le phénotype, on ne peut qu'être effaré par la catégorisation qu'a effectuée la Commission européenne sur la cisgénèse, l'intragénèse, qui a disparu d'ailleurs et qui est devenue un pool de sélectionneurs. Il est tout à fait illusoire, pour ne pas dire fallacieux, de vouloir regrouper des génomes sous des termes chapeaux, comme la cisgénèse ou l'intragénèse. C'est donner l'impression par des dénominations simplificatrices erronées que tout est sous contrôle. Les modifications de génome dans un individu peuvent avoir des conséquences différentes chez d'autres individus de la même sous-espèce et même du même pool de sélectionneurs. Le simple transfert de séquences entre variétés est en soi difficile et sujet à de nombreux effets non intentionnels. Donc vous avez déjà eu une idée avec les exposés de Sarah et de Francisca. Dans la diapositive suivante, je vous rappelle que la mutagénèse est un long processus qui a commencé dans les années 70. On a commencé les premières mutagénèses dirigées et dès la fin des années 80, on a commencé à développer de nombreuses promesses sur les techniques de mutagénèse dirigée sur des séquences. De nombreux candidats ont été proposés au cours de ces décennies. Et même si actuellement CRISPR-Cas est le modèle à la mode, on reste en fait toujours sur notre faim, car il existe de nombreuses combinaisons entre gènes aux effets inattendus et avec de nombreuses imperfections dans les outils qui permettent non seulement d'avoir des effets non intentionnels, mais qui en fait, n'élimine pas la prévalence du fond génétique. Sur la diapositive suivante, je vous rappelle que les principaux systèmes, deux des principaux systèmes dont a déjà parlé Sarah tout à l'heure, mais que le système le plus efficace reste encore le système d'Agrobacterium. La délivrance des réactifs CRISPR-Cas9 s'effectue sous différentes formes, sous forme d'ADN, d'ARN messager ou de protéines, les ribonucléoprotéines, qui sont toutes plus ou moins purifiées à partir d'organismes vivants. Tous ces réactifs sont contaminés par de l'ADN étranger, qui peut se retrouver dans les génomes des cellules transformées. Mais que par hubris ou méconnaissance, la majorité des expérimentateurs ne recherchent pas. Par ailleurs, les nucléases de type Cas sont censées éradiquer, c'est l'évolution qui a sélectionné ces Cas pour éradiquer, je dis bien, tout ADN ou ARN étranger. Il faut donc contrôler leur activité de façon à réduire le plus possible les durées d'activité pour essayer de réduire les modifications on ou off-target non intentionnelles. Donc, il faut se rappeler que ces techniques connexes apportent un certain nombre de mutations, d'épimutations non intentionnelles, comme le montre la diapositive suivante. Toutes ces modifications non intentionnelles, qu'elles soient on-target ou off-target, sont soi-disant éliminées au cours des rétrocroisements qui doivent permettre l'introgression du trait intéressant vers les variétés élites. Or, ces rétrocroisements sont loin, même statistiquement, sont loin de pouvoir éliminer toutes ces modifications non intentionnelles. Donc, on retrouve dans les variétés élites, pour de nombreuses raisons biologiques que je ne vais pas développer ici, on retrouve un certain nombre de cicatrices, tant des techniques connexes que des techniques NGT, et ces cicatrices dans les variétés élites se retrouvent en partie dans les cultivars et peuvent donc servir à la traçabilité. Sur la diapositive suivante, je rappelle deux problèmes qu'il y a avec CRISPR-Cas, c'est premièrement que les ARN guides sont issus d'une fusion de 2 ARN, et donc que cela pose beaucoup de problèmes de thermodynamique, de longueur, etc. Et donc ces effets off-target, etc. Et surtout aussi, le deuxième point, c'est la nécessité de séquences PAM pour l'accrochage de ces Cas. Or, avec l'usage des codons qui varient au sein du noyau d'une seule cellule, cela veut dire que pour pouvoir modifier des séquences au niveau d'un noyau, on sera souvent obligé soit de changer de Cas selon les séquences que l'on veut modifier, soit d'utiliser des systèmes beaucoup plus... Ces systèmes sont-ils, diapositive suivante, précis?
Excusez-moi, Yves, est-ce que je peux te demander de venir aux conclusions,
Oui.
s'il vous plaît?
Alors voilà une capture de l'attention, juste. Pour pouvoir vous dire ces effets non intentionnels. Il est en emboîtant, en allant à la conclusion, donc si nous peut aller à la conclusion, si les NGT empruntent la conclusion, les NGT empruntent le chemin des OGM transgéniques. C'est une dépendance au sentier assez classique. Pourtant, d'autres voies plus durables et à moindre impact sur l'environnement sont disponibles, comme l'holobionte, l'usage de variétés en mélange, ou la plasticité phénotypique, c'est-à-dire une meilleure compréhension du fond génétique. L'économie de la promesse des NGT ne peut conduire qu'à un verrou technologique qui capture les fonds de recherche publics et privés pour assurer ensuite les rentes des subsides de la politique agricole commune. Il est assez étonnant de savoir que des documents récents internes à la Commission démontreraient que cette Commission a désavoué ses experts de l'EFSA pour satisfaire les desiderata d'industriels. L'absence de véritable base scientifique à la proposition de la Commission d'exempter du statut d'OGM certains produits NGT induit une crise accrue de l'expertise et induira un désintérêt croissant des peuples européens envers des politiques au service d'enjeux socio-économiques internationaux, ne respectant pas les libertés de choix des producteurs et des consommateurs européens. Les agences européennes d'évaluation des risques et le Trent Research Center avaient déjà été pris à partie pour des conflits d'intérêts, des manques d'intégrité et la participation d'industriels à la rédaction d'avis. Mais les récents développements ont toute crédibilité à la Commission qui assurait jusqu'à présent ne se baser que sur des avis scientifiques. En conclusion, les NGT sont un pari risqué, basé sur un passif qui n'a pas fait montre de capacité d'adaptation aux changements en cours, tant sociétaux, avec la demande citoyenne de réduction des impacts de l'agriculture, que climatiques. Je vous remercie et vous présente mes excuses pour cet exposé trop long.
Mais non, Yves, merci beaucoup pour cet exposé très intéressant. Thank you very much for this very interesting presentation. And we're now going to go to the panel discussion, where there will also be time for everybody in the room to ask questions. I will just shortly present our speakers for the panel. So first we have Ana Afonso, who is a team leader for emerging risks at EFSA. It's not true anymore? Okay, thank you.
No, it's still me. I'm Ana Afonso, but I'm the head of the unit on nutrition and food innovation, which we support the GMO panel. I think I need to update my LinkedIn. I definitely need to do that.
Okay, thank you for correcting me. Then we have Martin Häusling. I think there I will not make a mistake. I know who he is. He is a long-standing Green MEP from Germany, and he is also the Greens shadow on the NGT file in the ENVI committee. And then Franziska Koller, unfortunately, she had to leave for an urgent commitment, but we will have Christoph Thehn who will be replacing her. He is the director of Test Biotech, co-founder and director. But I will first give the word to Ana Afonso for 5 minutes to have a short reply to what was presented. Thank you.
Thank you very much, Astrid. If you allow me, I would just actually go through a very short presentation. I'll try and do it as quickly as possible to just illustrate the work of EFSA. Do I need to ask for next? Okay, thank you very much. All right, I'll try and be brief, just to let you know a little bit where we stand. I think most of you do know. So EFSA is the scientific advice authority on the area of food safety. And we have been working on genetically modified organisms from the time we were created. So nothing new there. This is the current regulatory framework to which we follow on our assessment. Just a little thing about what is a GMO. Is an organism in which the genetic material has been altered in a way that does not occur naturally by mating or natural recombination. And this is where we stand for. Just to let you know what we've been doing so far. So far we have received 190 applications. This in fact corresponds to 83 events in basically 5 crops. This is what we're talking about. From those applications, 20 were received for future cultivation. And in fact, I haven't seen any since I took position because there hasn't been any application for cultivation for the last 10 years. From those, only 1 has been adopted and authorized, and that's maize. And as you know, it's only cultivated in 2 countries in Europe. For import and processing, we have done 170 applications. We're currently working on 35 of those. We have 5 inconclusive opinions. And it's important to explain to you what it means, inconclusive. Inconclusive means that there hasn't been sufficient data to conclude on the existence or inexistence of risks. However, we have adopted so far 113 applications. This means that for 113 applications, the EFSA GMO panel has considered there are no risks for the safety of humans, animals, or the environment. Okay, so I think you learned a lot about the differences between established versus new genomic techniques. And I'm sure my colleagues are far more prepared than I am to go through all the details and the complicated language. But just to explain you a little bit how this correlates with what we have as a legislation. So established genomic techniques basically means anything that was developed before 2001. So that's 22 years ago. And you heard about these names, agrobacterium transformation, gene-gun bombardment, and a few others that Sarah was kind enough to list. But these are a few examples. They're just techniques in which a transgene, out of that plant gene, is inserted into the plant. What's the characteristics? There's transgene expression, so there's something which wasn't there that has been introduced. There's a random integration, and Sarah went quite a lot in detail in explaining how that happened, but also some other speakers. And there's really no control as to the landing site for this transgene. Our regulation actually has some exceptions, and these are breeding techniques which are not considered GMO, and these are listed in part 2 of the annex of the current directive, but also in annex 1B. And these are UV, I'm giving again a few examples, UV irradiations, mutagenesis, cell fusion. So what is different about those? The techniques that develop them, there's no transgene expression, so any changes that are caused, there's no gene introduction, but there's random mutagenesis, which for most of you which are familiar with the area, you know that plant genomes are actually very fluid. They change, as all the other organisms. NGTs after 2001, you've been listening a lot about CRISP, and of course this is what everybody knows. It was a Nobel Prize in 2020? Yes. But in fact, under new genomic techniques, we talk also about other techniques, and the generic term I learned is site-directed nuclease. What does it mean for the NGTs? There's nothing exogenous, so we're not bringing something from out. There's no transgene expression, and there's an increased target specification and precision. Again, something that was well discussed before me. Okay, this is my dummy scheme for what it means, established genomic techniques and new genomic techniques. And I think we need to distinguish, because it's important to make this distinction between the techniques and the products that result from it. Do we have a transgenesis, cis or intragenesis? Do we have, and make well understand what are the differences. Same techniques can develop both kinds of products. Transgenesis, when you have the insertion of an exogenous gene, or not. You can use the same techniques to have just a modification on the DNA that already exists in the gene pool. What else do I want to say here? Nothing more, so I can go fast. What were we asked to do? So just to remind you, what we do as EFSA, we receive a mandate. So it's a question from the Commission. Can you please assess this application? Is it safe? We can also receive a generic question, which is for scientific or technical advice. And we've been doing work on new genomic techniques since 2012, but very recently we were asked in 2022 to update a former opinion of 2012. And what we were asked was certain terms of reference, I'll explain later, but just to remind you what it means, cisgenesis and intragenesis. So again, no transgene, but when there is an exact copy of the gene, we call this cisgenesis. When there is a rearranged copy of the genes, they are called intragenesis. Just a quick reminder. What we were asked was to identify potential risks for human, animal health, and the environment, comparing to conventional breeding and established genomic techniques. And also to determine whether the existing guidelines that we currently have for the risk assessment are adequate and sufficient, or whether there is a need for updated guidance. We were not asked for political implications, ethical or socio-economical considerations. That's out of EFSA's remit. We stick to the scientific. This work is conducted generally by a working group that prepares a draft report. This draft report is reviewed and finally adopted by a scientific panel of experts, which are carefully selected and independent, with a very strong policy of independency that EFSA as an institution follows. So no correlation with industry or any other possible conflicts, or so we do our best to ensure. Okay, what the opinion came out to was to say that no new risks are identified in cisgenic and intragenic plants compared with plants obtained with conventional breeding or established genomic techniques. No new hazards in cisgenic plants. Remember, the cisgenic was when we do not rearrange that copy. But additional hazards may arise for intragenic plants, which in fact is what is happening already. We also concluded that on a case-by-case basis, lesser amounts of data may be needed for the risk assessment of cisgenic and intragenic plants, developed by established or new genomic techniques. Current guidelines for assessing plants are partially applicable and sufficient. When we say partially applicable, is because, in fact, we recognize certain difficulties on the assessment. And then we were asked for a second piece of work, which was looking at, can we define criteria for risk assessment of plants produced through target mutagenesis, genesis, and intragenesis. We did that as a statement of EFSA, which is a work that is provided for advice for the scientific committee, but it also indicates that there's further work to be developed in certain domains. But that it was this criteria that the European Commission took over to start drafting some of the scientific considerations on the current regulatory proposal. I have my face on top of the criteria, so I'm serious. I can show you later, but I think you know that. But basically, we are looking at a first step. Is there exogenous DNA present? And I think you can recognize this terminology as well in the so-called category 1. Is the DNA sequence from the breeder's gene pool? What does it mean, breeder's gene pool? Plants that can mate naturally, so that they are of the same kind. What is the type of integration? And this is very much important. It has also some resonance to some of the aspects that were already mentioned. And all this provide the equivalence to what happens in nature, but also in conventionally breeding plants. And then we talked about unintended interruption of an endogenous gene, and that's criteria number 4. And if we go on our decision tree, we will look at history of use or familiarity, depending on which context you're looking at it, and the function and structure associated to the new allele that was introduced. And I think this was what I wanted to say, just to say that breeding techniques will continue to develop. We've been working on breeding techniques for as long as humans started farming. And in fact, what we have as plants now and what we are cultivating is very different from what it was in the past. We're really talking about very few species, and I think biodiversity was something that was talked about, a subject which is independent of this opinion as such. What we also see is that there is the need to work on different traits, and I have to, as a risk assessor, have to say that currently, with established genomic techniques, or even in conventional breeding, there are traits which are difficult to assess in risk assessment, and EFSA is always working to be prepared and further develop. What I would like to say is, independently of the decisions that are taken on the political level, we will continue to work and expand the details on what we have published as criteria, because these are important in the risk assessment. But if this regulatory proposal is adopted, of course, EFSA will be working on future mandates, on developing data requirements, and for the risk assessment of these plants. Thank you so much, Astrid. I hope I was fast.
Thank you for these very interesting explanations of your work. We will now move to Martin Häusling. Maybe you can explain to us, how you're going to use this information which was presented today in your political work. I would be interested.
So thank you very much for the invitation, Christoph. I want to make my speech in German. We have translation. Ja, ganz herzlichen Dank für die Einladung. Und also ich bin ja jetzt schon ein paar Jahre in der Politik. Ich habe auch schon in den 2000er Jahren die Diskussion verfolgt, damals die großen Versprechen der Gentechnik, der alten Gentechnik, muss man jetzt sagen. Und was ist daraus geworden? 2 Arten von Pflanzen, die einen, die selber Gift produzieren, und die anderen, die Resistenzen gegen Pestizide haben. Viel mehr ist ja nicht passiert in den ganzen Jahren. Und jetzt kommt die neue Welle mit CRISPR-Cas, und jetzt tut sich eine Wundertüte auf. Und jetzt können wir alle Probleme, die wir haben, der Welt lösen, vor allem klimaangepasste Pflanzen. Wenn ich mir das jetzt anschaue, was ist auf dem Markt und was hat Marktreife, ja, dann haben wir die GABA-Tomate, die blutdrucksenkende Tomate. Ich glaube, darauf hat die Welt gewartet, das muss man mal deutlich sagen. Der Bayer will einen Mais auf den Markt bringen, der kürzer ist und deshalb nicht so anfällig ist für Schäden durch Wind. Das sage ich nur als Biobetrieb, als Biobauer schaffe ich das auch, so Mais nicht so groß zu kriegen, da brauche ich keine Gentechnik für. Also es werden jetzt Wunderversprechen gemacht, die nicht haltbar sind. Und alle Diskussionen, die jetzt im Moment ablaufen, konzentrieren sich sagen, auf Kennzeichnungen und auf Patente. Beides hat eine enorme Wichtigkeit, keine Frage. Aber keiner redet mehr darüber, dass wir eine Risikoprüfung brauchen. Ich glaube, die Vorträge haben heute Morgen sehr deutlich gezeigt, es gibt ein Risiko. Es gibt ein Risiko und dann schauen wir nur mal, was Frau Carpentier, die das ja erfunden hat. Gibt es keine Übersetzungsprobleme? Is there no translation?
There was a problem with translation, but it came back.
Yeah, we're
Okay.
having interruptions.
Okay, it came back. So, Frau Carpentier hat, die den Nobelpreis 2020 bekommen hat, ja selber davor gewarnt und hat gesagt, es ist eine sehr gefährliche Technik. Und in Deutschland hat der Ethikrat ja ein Moratorium bei dem Einsatz von CRISPR-Cas bei Menschen ausgesprochen. Und im humanen Bereich sagt man, nee, das ist zu gefährlich, bitte nicht auf CRISPR-Cas setzen. Es gibt Risiken, die wir in dem humanen Bereich nicht absehen können und die wir nicht wollen. Aber jetzt wird natürlich gesagt, im Bereich der Tiere und der Pflanzen, ja, da können wir ja alles Mögliche probieren jetzt. Warum dieser Unterschied? Sind Tiere nicht sehr nah verwandt mit uns? Warum macht man diese Unterschiede jetzt? Und ich finde, muss ich auch noch mal sagen, die Vorlage der Kommission ist keine wissenschaftliche Vorlage, es ist eine politische Vorlage. Man will etwas durchsetzen, was man politisch erreichen will. Und man hat sich ja, das haben wir ja schon kritisiert, ganz am Anfang auch wirklich darauf konzentriert, dass bei den Anhörungen in der Kommission die Verteilung derjenigen, die da Stellungnahmen gemacht haben, sehr einseitig war. Ja? Wer hat denn da in erster Linie bei 70% der Anhörungen bei der Kommission vorgesprochen, nämlich die Industrie? Und das hat man 1:1 übernommen. Nur mal eine Frage dann an die Kommission. Wie kommen Sie eigentlich auf 20 gentechnische Veränderungen einer Pflanze und die könnte man, das war ohne Risiko? Warum nicht 15? Warum nicht 30? Warum nicht eine? Allein das zeigt doch schon, dass eine rein politische Botschaft ausgesendet wird. Man muss sich da mal vorstellen, 20 einer Pflanze, aber was ist, wenn man die Pflanzen miteinander kreuzt? Wie viele gentechnische Veränderungen haben wir denn dann? Ja, überhaupt nicht berücksichtigt in der ganzen Vorlage. Und dass diese Heilsversprechen, die man da macht, wie gesagt, komme aus einer langen Diskussion um Gentechnik. Es gab mal in Deutschland die Entwicklung einer Kartoffel am Flora. Die sollte dann bestimmte Eigenschaften haben bei Stärke, damit sie besser verarbeitet werden kann. Ja, ganz groß gehypt, damals von BASF. Das Problem war nur, die hatte zwar die Eigenschaften, sie hatte nur keinen Ertrag. Und deshalb kann man ganz klar sehen, wenn ich einen Teil in einer Pflanze umbaue oder im Tier, kann das Auswirkungen haben auf ganz viele Faktoren. Wenn ich eine Pflanze haben möchte, die weniger Wasser braucht, schön, ja, aber hat sie dann noch Ertrag? Oder was hat sie ansonsten für Veränderungen? Also zu glauben, und das finde ich, ist auch der Fehler in der Kommission, man tut so, als müssten wir beim Computer hier und da was ändern und dann kriege ich ein neues Programm abgespult und schon haben wir das gewünschte Ergebnis. So einfach ist es halt nicht. Und das muss man auch mal deutlich sagen. Und was kommt auf den Markt? Das, was jetzt vorliegt, was auch Sie eben vorgestellt haben, zeigt ja deutlich, es geht gar nicht um klimaresistente Pflanzen. Was heißt eigentlich Klimaresistenz? Wir haben ja dieses Jahr gesehen, wir hatten erst Trockenphasen, dann hat es ganz fürchterlich geregnet. Ja, was für Pflanzen müssen wir denn jetzt entwickeln? Also wasserresistente oder trockenresistente Pflanzen, das kann man überhaupt nicht abschätzen. Und deshalb ist die Frage, was kommt auf den Markt? Auf den Markt werden Designerpflanzen kommen. Es wird um Designerpflanzen geben, die vielleicht besser zu verarbeiten sind, die bestimmte Bereiche betreffen. Und da stellt sich halt die Frage, was ist zum Beispiel, wenn jemand dann Allergien hat, wird das berücksichtigt, wenn man solche Sachen auf den Markt bringt beim menschlichen Essen? Und das sind alles Sachen, die muss doch eine Risikobewertung vorgeschaltet sein. Man kann nicht einfach irgendwas auf den Markt bringen und sagen, warten wir mal ab. Das Zweite ist, was ist eigentlich, wenn man dann tatsächlich merkt, und selbst die Kategorie 2, muss ich auch noch mal deutlich sagen, die hat wirklich auch keine konsequente Risikovorsorge, sondern das kann die Kommission entscheiden. Hat eine Pflanze ein Risiko? Also auch Kategorie 2 ist weitgehend befreit von Auflagen. Das muss man auch mal deutlich sagen. Aber was ist eigentlich, wenn die Kommission oder andere feststellen, die Pflanze, sorry, die ist aus dem Ruder gelaufen, die ist nicht so, wie wir das alle gedacht haben. Und dann kommt das große Risiko, was wir dann haben. Ich kann bei der roten Gentechnik einen Deckel drauf machen, kann sagen, okay. Aber was ist, wenn wir Pflanzen, die dann massenhaft angebaut werden, wir merken, es gibt sozusagen Folgen für die Umwelt. Wie holen wir die zurück? Also einmal freigesetzt, wie holen wir diese Pflanzen zurück? Und das ist doch das Risiko, was wir haben, was man ganz deutlich auch sagen muss. Da gibt es keinen Irrtum und Irrtumkorrigierung, sondern da gibt es tatsächlich dieses Risiko für die Umwelt, was wir dann überhaupt nicht mehr einschätzen können. Ich erinnere nur daran an diesen gentechnisch veränderten Lachs aus Kanada. Was ist, wenn solche Tiere in die Umwelt kommen? Wie verändert sich dann eine Lachspopulation dann zum Beispiel? Wandern die dann noch oder werden dann alle so groß? Also das sind Risiken, die muss man doch abschätzen. Nur noch mal kurz, weil du schon so kritisch guckst. Ich muss leider jetzt weg. Wir haben nämlich heute im Agrarausschuss die erste Diskussion. Und was hier gerade passiert im Parlament, ist eigentlich ein Skandal. Ich sage es mal ganz offen. Gestern haben die Koordinatoren der anderen Fraktionen beschlossen, wir beschleunigen das ganze gesetzgeberische Verfahren. Es soll jetzt in einem Tempo durchgezogen werden, solange die Öffentlichkeit nicht merkt, was da gerade geplant wird, soll jetzt schon in einer relativ kurzen Frist bis am 11. Dezember sollen wir abschließend beraten im Agrarausschuss und eine Woche später die Stellungnahme des Umweltausschusses. Und dann soll es im Januar durchs Parlament gehen und im Februar sollen die Triloge anfangen. Am besten alles noch vor den Wahlen, damit keiner merkt, was da gerade für eine, ich sag's mal auf Deutsch, Sauerei läuft. Und das finde ich ein Skandal, dass man hier auch noch die Normen des Parlaments außer Kraft setzt, nur um das hier schnell mal durchzuziehen. Solange die Öffentlichkeit nicht darüber diskutiert, läuft das alles unter dem Radar. Das finde ich in Anschlag auf unser Verfahren. Ja? Und dass das also von den großen Fraktionen dann noch getragen wird, halte ich für untragbar. Und dass die Kommission dieses Spielchen da mitmacht und Märchen erzählt, haben Sie schon mal den schönen Werbefilm gesehen von der Kommission zur neuen Gentechnik? Das ist Bullabü, das könnte von Bayer BASF stammen. Ja? Das sind wirklich keine seriösen Diskussionen, die wir da gerade führen. Und ich hoffe, Christoph, wir können und müssen da noch was korrigieren im Rahmen der Anhörung im Umweltausschuss. Nochmal heute, wenn Sie jetzt erwarten, dass heute die Berichterstatterin im Agrarausschuss soll heute ihren Bericht vorlegen und dann darüber diskutieren. Die Berichterstatterin hat doch gar keinen Bericht. Sie hat doch keinen Bericht, über den wir heute diskutieren. Wir sollen jetzt Änderungsanträge machen. Wir wissen aber gar nicht, was die Berichterstatterin sagt. Also ich bin ja schon ein paar Tage im Parlament, aber sowas habe ich nicht erlebt. Und das Ganze unter der Farm-to-Fork-Strategie, die ansonsten hoch gelobt wird, da muss ich sagen, da bin ich als Parlamentarier entsetzt. Bin auch entsetzt über die Unwissenschaftlichkeit der Kommission und dass EFSA dann noch den Segen dazu gibt, das setzt im Ganzen dann noch mal eins drauf, eine Krone auf. In dem Sinne muss ich jetzt wirklich in den Ausschuss, um mich da auch noch mal aufzuregen. Ich bedanke mich für die Einladung und wir werden bestimmt noch weitere Veranstaltungen haben, um auf eins zu machen, Öffentlichkeit herstellen. Wir müssen die Öffentlichkeit über das informieren, was da gerade läuft. Es ist nicht nur eine Bedrohung des Biolandbaus, sondern letztendlich auch eine Bedrohung unserer Verbraucherrechte und dessen, was wir im was wir in Europarechten haben, ein Vorsorgeprinzip. Das wird einfach über Bord geworfen. Vielen Dank.
Thank you very much as well, Martin, for joining us here today. And we now have only 10 minutes left for the final discussion. Christophe?
Moi, je voudrais juste poser 3 questions, notamment à l'EFSA, mais ça peut amener des réponses des autres acteurs. D'abord, je voudrais saluer la présence de mon collègue, M. Lecas, qui est le shadow socialiste en commission Agri. Donc, merci beaucoup d'être là ce matin. J'avais 3 questions. Dans la proposition de la Commission, il y a la possibilité en catégorie 1 de présenter un dossier de nouveaux OGM pour les essais en plein champ et ensuite avoir une autre forme d'autorisation automatique pour la mise en culture sans que ça passe par l'EFSA. Et par ailleurs, il y a des conditions qui sont très dures pour les autres États pour poser une question dans le processus d'autorisation quand le processus passe par l'EFSA. Et dans la proposition de la rapporteure, elle considère qu'il faudra que les États aient des arguments justifiés, fondés, pas des commentaires, mais des arguments. Donc, est-ce que vous n'avez pas la crainte que finalement, l'EFSA ne voit passer quasiment aucun dossier de NBT en catégorie 1? Ça, c'est ma première question. La deuxième question, c'est j'ai cru comprendre dans votre diapo que vous disiez que vous n'aviez pas identifié de risques nouveaux par rapport au conventional breeding et par rapport aux OGM passés. Si c'est bien ça que vous avez dit, ça veut dire que ce n'est pas ce que dit souvent la Commission, qu'il n'y a pas de risque supplémentaire par rapport au conventional breeding. Et puis, dernier élément, sur le gene pool of a breeder, vous avez dit « can occur ». Et comment vous, dans la proposition de la Commission, il est dit que le gene pool of a breeder, c'est tout ce qui peut se passer dans la nature. Mais à certains moments, est-ce que c'est could, est-ce que c'est can, au sens où est-ce qu'il faut avoir vérifié que ça s'est déjà passé dans la nature? Et donc du coup, vous, EFSA, quand vous regardez les risques pour l'environnement, est-ce que vous regardez par rapport à ce qui se passe réellement dans la nature ou par rapport à des modèles théoriques de ce qui pourrait se passer suite à des rencontres possibles? Je ne sais pas si c'est très très clair ce que je dis, mais il y a deux visions du gene pool of a breeder de ce point de vue-là.
Thank you very much. I think those questions were mainly directed to EFSA. Maybe we can take one or two more questions from the floor, and then we move to the panel to give the responses. Please, Eric, go ahead.
Yes, thank you. I'm Eric Gall from Inform Organics Europe. I would like to hear from Sarah or from Yves. What are the prospects for the development of detection methods for NGTs, especially given the numerous unintended effects that were mentioned as well? Thank you.
Yes, detection, a very important issue, especially for the organic sector, because otherwise how are you going to make sure that this will not crossbreed? I think, Benny, you also raised your hand.
I'd like to ask Madame Afonso, could you kindly respond to the concerns that you have heard from the scientific colleagues before, and not just repeat the procedure of the EFSA? We need a discussion about these risks, and not you tell us one thing about processes and the others tell us something about risks, and you just don't respond to it.
Thank you for your critical comments. Okay, let's take these two last questions, but then we really have to move to the panel.
Hi, my name is Zelda Skrammer from EU Action Group, and I've got two questions to Anna. She mentioned that there are no additional risks to humans as well as to animals. I would like to know, first question, what are the risks that have already been identified on GMOs to humans and animals? And then the second question is, there are already well-documented and studied unexpected effects and health risks posed by genetic engineering to humans, such as toxicity, loss of nutrition, cancer, antibiotic resistance, allergies, and immunosuppression. Anna, I'd like to know what long-term studies support your assertion that no risks have been identified in addition to existing risks.
Right. Hello, my name is Elix Germa, and I have a question for the lady of EFSA. You specifically say that intragenesis and cisgenesis, there is no, there's basically no risk. You said that there's not a risk category for compared to metagenesis. However, can you guarantee that there is no risk in accidental genetic damage when you start copying cells, or when you start using modifying cells with the genetic material of the same plant, for instance, or the same animal in this case?
Thank you for your questions. I'm sorry I cannot take more questions because the time is really restricted. I think we could talk about this topic for 1, 2, 3 hours, but unfortunately we have 5 minutes left. So I would like to, if the two of you want to respond, I would like to give 2 minutes to each of you, and then I would like EFSA to have the final word until we really have to switch to the next panel. Who would like to start?
Yes, thanks for the question on detection. I was actually reading again the proposal, and I find on page 22 this text, which refers to detection. The European Union Reference Laboratory for GM Food and Feed, in a collaboration with the European Network of GM Laboratories, concluded that analytical testing is not considered feasible. And now we just recently got the European Commission to invest 10 million euros on detection methods under the Horizon Europe research program, to which many of these members are in the consortium. So what is written in the proposal does not correlate to what's happening in the network. We are developing methods. The Commission is funding the development of these methods. And if there isn't methods yet, it's because there was no research funding for this. And now we will have 3 years of research funding. But there is a team here in Belgium, in Cienzano. They have published a lot on gene-edited rice, gene-edited microorganisms. We have a project running from the Norwegian Research Council since 2020. We have published 2 papers. We are now coming to 2 others on detection methods, so it's evolving. The topic is evolving. If I can just briefly make another comment, and I think that reflects the reaction of the audience to Anna's presentation and EFSA work. It's also my impression that I see a gap between EFSA and what is in the proposal. For example, when EFSA states case-by-case risk assessment, that's not reflected in this Annex 1 category. It's not case-by-case, it's structured and fixed criteria. And also the equivalence, when there is this loose thresholds, that's also space for risk to emerge in these criteria, because they won't be equivalent if they can be different up to 20 technical cements nucleotides. So maybe this gap is what also burns about in our heads. Thank you so much.
So very shortly, if we look to the presentation we've heard from Franziska Koller, there's now already 50 applications going on in the family of oil plants, Brassicacea. And of course, when you look to these applications, there will be many intended and unintended effects which are unlikely to occur with conventional breeding. That's for sure, if you look to the data. So it's very clear we need data. What are these differences? What kind of effects have to be expected? What about cumulative aspects if you release them to the environment and they cross or get stacked by purpose? And the current proposal of the Commission makes it impossible to get this data. There are no data requested case by case. They're only looking for very general criteria. So what we really need to get rid of is a formal catalog of criteria that is supposed to establish equivalence. You have to go back to a system which establishes similarity or equivalence, whatever you mean, by case, by really molecular data, by molecular risk assessment. And so if we may use the flexibility within the current system, there is a step-by-step procedure. So on the first step, we have the molecular risk assessment, molecular characterization, and this could be expanded in a way that we really can have more certainty about the equivalence or the difference between these plants, NGT plants and the plants derived from conventional breeding. And if we are not using this data, not using this flexibility, we'll end up with loss of control, with irreversible releases, new problems for the next generations, and some of the decisions we take now on this question cannot be reversed again as soon as we release these plants without risk assessment to the environment. We are the responsible generation to take this decision now, and the Parliament is really obliged to carefully address these issues in its final opinion. Thank you very much.
Thank you. So I give the final words to Anna. I know there were a lot of questions asked to you in specific, but still, if I may ask you to be as short as possible, because we have very little time left.
I will do my best. How much time?
nothing really, but...
That was easy. So, Monsieur Clergeau, the first question, I will leave it to my colleagues in the European Commission, because I think you were asking specifics of the draft proposal. They know it much better, and also for the changes proposed by the Parliament rapporteur. So I will leave that one. The second question, if I recall correctly, was... let me just take my notes, and I'm sorry, I was trying to follow. But it was about comparative assessment, and this is done so far. I think what we should remember, and about the EFSA's opinion, and I'm not going to discuss the proposal because we are not responsible for drafting legislation, we just do the science. The questions were, is it comparable to conventional breeding? And I may recall you as well of the exceptions that currently exist in the legislation, in the annexes of the legislation that are exempt of being treated under the risk assessment process. So yes, in terms of, you were also asking about can breeders pool? And the question was, how do we do that? How do we do that? That's exactly what we do already. When we're looking at a comparative study, we're looking at what exists in the nature. I would like to remind you all, and this was said several times before, that there is a fluidity in the genome of a plant, as there is in any of our genomes. We change, there are changes. So that is important, but that is, the comparison is being made. There is some discussion about gene pool brothers, and there's also other institutions in the European Union that guarantee that those varieties are properly catalogued, and work will be done as well with those institutions, I'm sure, if this proposal comes forward. There was a comment from Euractiv about health risks, so consequences of CRISPR-Cas utilization in human genome. I'm sorry, I cannot comment on that. What we look at is the risks for humans on consuming such varieties, such foods. Okay, so it's a quite different kind of work, and I would abstain from commenting about that. Unintended effects, and the gentleman asked, why don't we answer? We did, in many occasions. I have to notice that the paper that was presented by the colleague of Christophe is actually a review paper summarizing information that has been scrutinized by the EFSA GMO panel in several occasions. So even in the little time I had available, I tried to illustrate what we do as EFSA, but of course, please, I would be more than happy to provide detailed information about each of the arguments that were presented, which are not new to us. Unintended effects is something that we look at in the current framework, and all the different papers that have been cited, or in this paper, that, as I said, is a review, is not a primary research, have been carefully scrutinized by all our experts. And I think I've done it.
You did a very good job.
Thank you so much, and thank you for the invitation.
So thank you so much for listening to this first panel, and we will now switch directly to the second panel, which will be about consumer choice, and Christophe Clergeau will be chairing it. Thank you.
There is no specific plan, as you want. Bon, merci à toutes et à tous. Je vais poursuivre en français. Donc, nous avons eu l'éclairage scientifique. Nous sommes habitués, quand on parle de ces enjeux-là, à avoir des controverses scientifiques et des débats sur la manière dont la science est prise en compte dans le travail fait par l'EFSA et dans ensuite la prise de décision politique. Et ce sur quoi nous allons nous confronter maintenant, c'est comment est-ce que l'on peut penser le processus de décision politique, c'est-à-dire comment prendre en compte l'ensemble de ces informations dans la définition de la législation et dans la définition des modes de décision, en entendant deux types de paroles. La parole de chercheurs qui travaillent sur ces questions de gouvernance, de l'innovation, de l'expertise, de construction des processus de décision politique, et puis la parole d'acteurs de la société. Donc je remercie les représentants, je reviendrai à une présentation plus détaillée des consommateurs et des producteurs bio, pour qu'on puisse aussi discuter de la manière, ce que j'évoquais au début de cette conférence, dont les droits citoyens des consommateurs et des producteurs sont pris en compte ou pas dans la définition de la législation, pour des raisons qui peuvent être à la fois des raisons scientifiques et à la fois des raisons éthiques, de conception même de la société, de son organisation économique et de ce que les uns et les autres peuvent prendre comme décision. Alors, on va commencer par des éclairages de chercheurs sur les enjeux de gouvernance, de l'innovation, de l'évaluation des risques et de la prise de décision politique. Et donc, je vais donner la parole en premier à Jeroen van der Swisch, qui est professeur à l'université de Bergen, donc deuxième intervenant norvégien, comme quoi la Norvège est de plein pied dans tout le débat scientifique et sur l'innovation, qui va nous montrer comment on peut poser ce débat dans des termes politiques et éthiques et faire dialoguer le discours de promesse, qui est celui autour des OGM, et les attentes de la société et les enjeux de gestion des risques. Et ensuite, Hélène Vos reviendra sur les éléments plus juridiques de législation et la manière dont on peut mettre en dialogue ce qui est en débat aujourd'hui avec tous les débats des 20-30 dernières années autour des OGM et autour plus globalement de l'évaluation et de la gestion des risques. Et puis bien entendu, j'ai à mes côtés la Commission à qui on donnera le temps de répondre. Je pense qu'il y a eu la possibilité avec l'EFSA d'avoir des réponses complètes pour qu'on puisse être dans ce débat, dans ce dialogue. Donc d'abord, Monsieur Van der Swisch, qui est à distance. Donc j'espère que tout va bien fonctionner. You have the floor for 10 minutes.
Yeah. Thank you very much. Sorry that I couldn't make it to Brussels, and thanks a lot for allowing me to speak online. So I will talk about the role of the precautionary principle in dealing with new and emerging gene technologies. Can I have the next slide? So why do we have the precautionary principle, or it's also called the principle of foresight, or the anticipatory principle? And it starts, for instance, with the book The Social Control of Technology by David Collingridge, where he noted that the social consequences of a technology cannot be predicted early in the life of the technology. And by the time the undesirable consequences become obvious and clear, the technology is so much part of the whole economics and social fabric that its control is extremely difficult. So that's why we need to have foresight and anticipation. Another very relevant concept is from late Ulrich Beck's book Risk Society, where he explains how disasters like Chernobyl accident and the BSE mad cow disease crisis are the result of organized irresponsibility. And when I read the proposal, this was the word that came to my mind by just randomly excluding a whole group of class 1 edited plants. from any risk assessment, that's an act of organized irresponsibility that just puts away the precautionary principle. Can I have the next slide? So Ellen Vos will present the Recipes project, a big European project on the precautionary principle in Europe in the past 20 years. And one of the products that we developed was the sector-specific brief on the precautionary principle for gene technology in agriculture. And there are many good recommendations there that the Commission could have taken into account. The first is strengthen the application of the precautionary principle, both as a compass in technology development and as a safeguard for consumers, the environment, health, and other important values of Europe in avoiding harm by being proactive and anticipating risks. Then we have the Cartagena Protocol that does not have any exception for class 1 or 20 base pairs edits, but request for all edited or changed organisms a case-by-case risk assessment. It's just mandatory, so it's very strange that this proposal does not comply with the Cartagena Protocol. Then we have learned that it's important to have upstream societal engagement, also pre-research agenda setting. So why are we developing these technologies and what societal goals do we want to serve with it? And it means also promoting the precautionary principle in research. It also requires the possibility for post-authorization review and mechanisms of revocation, and these seem to be missing. We even see that there are some possibilities where member states can no longer issue national bans on CRISPR-edited plants. We also should allow for alternative research pathways to achieve the same objectives as we do with edited crops. For instance, agroecology can serve in a proven way and faster the same objectives. We also have to promote safety by design, be explicit about problem scoping, broaden the risk assessment, address conflicts, and also anticipate transboundary harm. The anticipation of transboundary harm seems also missing in the proposal. And then make power asymmetry explicit in the participatory processes. Can I have the next slide? So there are two very important reports by the European Environment Agency in Copenhagen. The first was in 2001, and then the second volume came in 2013. It's the late lessons from early warnings studies about precaution and innovation. And there are 12 lessons, and the slide here lists the 12 lessons. In view of the limited time, I just highlight a few that are most relevant in this case. And the first is provide adequate long-term monitoring so that early warnings are early picked up and acted upon. So we see here that category 1 and even many category 2 crops are exempted from monitoring of possible environmental effects. So there is no long-term monitoring, not even for all category 2 crops. Then number 5, ensure real-world conditions. In the previous session, we saw that many of these assumptions about precision, about predictability, are proven to be invalid. So the assumptions behind category 1 are not real world. So the lesson from the late lessons report is not followed up here. Then scrutinize the claimed justifications and benefits of these technologies. There's a lot of work to do, and I heard already some good points here in the previous discussion. And then also promote alternatives. Where is the investment in alternatives, in other technologies that meet the same needs? In organic agriculture, for example, or agroecology, where you do not go against but with nature and use ecological processes. And there is a lot of innovation there that should be stimulated. And then maintain regulatory independence is a crucial issue here. Can I have the next slide? Philosophers of technology have highlighted that the metaphors of CRISPR are highly misguiding. the metaphors like scissors, precision editing, editing the genetic code. CRISPR may soon become as reliable as a text editor. This is not consistent with what we know about the technology, and this is not something that CRISPR and other gene editing technologies deliver. And then reading in the European Commission questions and answer website that this technology allows precise and efficient development for improved plant varieties. That is very strange to read with the known state of technology. And also, who decides what is an improved plant variety? In what sense is it improved? Because plants can be good in many viewpoints, not only that they are drought resistant or have a high yield, but also that they are environmentally safe, not invasive, etc. So is it responsible to use such metaphors is something we have to reflect on, because describing the genetic system as if it is just electrical circuits where we can switch on and switch off things and can predict what happens, that is not the case with the genome. It's a very complex system, and from complex system biology, we know that even minor changes can have big consequences. We remember the coronavirus, and just a small mutation in the genome of the coronavirus, less than 20 base pairs, made the difference between lethal or benign, or between highly infectious or hardly infectious. So the current skewed metaphors can silence the negative aspects of technologies, and should not go beyond what is scientifically established. So don't keep reproducing these wrong metaphors. Can I have the next slide? So here we see, and this was already highlighted in the previous session, so I can go rapidly over this, but these enzymes like CRISPR-Cas, they cut also in other places than the places you have designed them to cut. And that has a lot of implications indeed. So what other damage might you have done at other sites in the genome? And we know from the concept of gene duplication, which is something that evolution has done over the past millions of years, to duplicate genes in complex organisms like plants and animals. That means that the same sequences of DNA are in many places in the genome, and we don't know the functions they have in other places than the target area. So there is a lot of unknowns in that area that is highly relevant for understanding the risks. Can I have the next slide? This was already mentioned by one of the previous speakers, but we know that when CRISPR cuts the double-strand DNA, which is the way it works, then the cell responds with its own defense mechanism. Also plant cells do that by repairing the DNA. And in this natural process of a plant or an animal cell to damage to its DNA, a lot can go wrong. And you get a lot of random and complex rearrangements, as this paper showed. And if I can have the next slide. So the key thing here is that we have to understand the DNA damage repair in plants after a cut of the double helix. What CRISPR does, what these genome editing technologies do. And the knowledge about the repair process of DNA that plants naturally have is in the very early stages. If we look at the number of published articles, it's only 10% of all the body of literature on this mechanism, because most of it is on humans and animals, and plants are just a blind spot in our knowledge. Can I have the next slide? Next slide, please. Yes. So we see that we have many types of uncertainties. We have the known knowns, the unknown knowns, the known unknowns, and the unknown unknowns. And in this domain, we must recognize that what we have is a dominance of the category unknown unknowns, where we don't know what we don't know. Can I have the next slide? So the point is we need more precaution for these new genomic technologies. We simply have a lack of history of safe use. This has previously been recognized also by the Court of Justice in an earlier court case. We have a lack of history of safe use for CRISPR and related technologies. And then we have to recognize that the hazards that are involved are not just risks, but they all involve uncertainties and ignorance. The damage control, if things go wrong, may completely fail, because comprehensive risk assessment, even if they were comprehensive, but that's not the current proposal for a large part of the proposed technology development, that is insufficient to guarantee damage control. And we don't know in which domain they will give damage. Will it be new invasive species? Will it give other problems like changing the position of organic farmers? It can have social implications. So, and then also the assumption of precision and predictability. And I think this was proven in the first session by the knowledge from the science. It is invalid. It is not precise, and the outcomes are not predictable, and that is independent of the number of base pairs changed. So the number 20 is not a prediction for that it will be safe. It can also be used to make cross-species insertions. So I heard the EFSA person saying that it's not exogenous, but that's just a word game. Because, okay, it's exogenous if you take out the word rat from a book on the rat genome and insert it in a friend's book on salad, in the word salad, and then you have cut and paste, and then it's exogenous because it comes from another book in another language with the same characters. But now you go into the book and type the word rat inside the salad, and it's suddenly no longer exogenous, but the result is the same. So it's just a word game.
Okay, excuse me, can you go to your conclusion, please?
Yes, can I have then the next slide? So what we need is more responsible research and innovation, where we ask the product questions: how will the risks and benefits be distributed? What other impacts can we anticipate? Not only the direct biological narrow type of risk, and what might we never know about? The process questions: how should standards be drawn up and applied? How should risks and benefits be defined? Who's in control? Who's taking part? And also, who will take responsibility if things go wrong? And there is not even monitoring, as I said. So how can we take responsibility, or how can we even detect timely that things go wrong? And then we also have to ask the purpose question as: why are researchers doing it? Are the motivations transparent? Are they in the public interest? Who will benefit? And what are they going to gain? And what are the alternatives? I think I end my presentation here, and thanks for your attention.
Thanks very much. Je vais revenir au français. Je crois que dans ce débat, nous avions, en tant que groupe socialiste, et d'autres aussi, plusieurs possibilités. La première, ça aurait été de dire non à la législation. La deuxième, ça pourrait être de dire il y a un risque réel de défaut d'évaluation des risques, voire de non-respect du principe de précaution. On regarde des incertitudes scientifiques dans un domaine où la science est nouvelle et où elle évolue très rapidement, et donc on finit à la Cour de justice. On verra. Mais aujourd'hui, notre approche n'est pas celle-là. Notre approche, c'est de faire une bonne législation et d'arriver à remettre dans cette législation les éléments qui apparaîtraient les plus importants pour prendre en compte ce qui a été dit par les scientifiques et ce que vont dire ensuite les représentants des consommateurs et des producteurs. Donc c'est très important pour nous, et je vais donner la parole à Hélène Vos, de remettre la proposition de la Commission dans la perspective un peu historique de la manière dont on a approché la régulation des risques autour de ces sujets alimentaires au cours des dernières décennies. Donc Hélène, qui est co-directeur du Centre européen juridique de l'Université de Maastricht, qui est sur ces sujets depuis longtemps, qui a récemment fait un travail pour l'EFSA sur l'indépendance de l'EFSA, qui a été présenté en commission Envie, donc qui est très familière de ces sujets. Vous avez la parole.
Okay, well, thank you very much. Thank you very much for the invitation to talk about this extremely important topic. And I have structured it in the way that I've sketched it out, and I will, without losing my minutes to explain the structure, I will immediately go into the detail. So let me go back a bit to the basics. Where do we come from in EU risk regulation? How is EU risk regulation structured? We all know that there's a risk assessment done by EU agencies, the members of the member states authorities. The risk management is done by the European Commission, and alone under a delegated act or together with a comitology under an implementing act. And there is then the risk communication to the general public by both of these actors, or all these actors. So these elements together, in fact, are risk assessment, risk management, and risk communications are forming the principle of risk analysis. And we all know that this is a historical perspective. This is coming from the BSE crisis, where there was a strong need to regain trust in EU food regulation, and the principle of risk analysis was inserted in the general food law as a general principle of food and feed law. So this was an important measure to regain public trust in the regaining of in the EU dealing with the risk and uncertainties. Now, the precautionary principle was at the same time also introduced in the general food law, and hereby codifying the Court of Justice case law, that the principle of the precautionary principle not only applied to the environment, but also applied to protect human health and safety. The principle of risk analysis we find then back in the different regulatory risk schemes on risk items, for example, some medicinal products, we see food, chemicals, whereby prior authorization procedures are considered as an expression of the precautionary principle. Now, my colleague Jeroen van der Sluis has already talked a bit about the precautionary principle. It's very much of a constitutional principle, and it is it's a principle that is defined here, as I said, I wrote here on the slide, already in the case law of the Court of Justice and codified in the general food law. It is not a static principle, really, but it's actually inviting for more knowledge acquisition and to carry out a more comprehensive risk assessment. It is also important that we conclude that the precautionary principle as such are not opposed. And this is something, in fact, that we very much investigated in the recipes project, that Horizon 2020 project that also Jeroen van der Sluis was a partner of, and we together in the whole consortium, we developed in this guidance on the application of the precautionary principle, a way of how to deal with, responsible deal with uncertain risk in the development and implementation of technology. Yeah, there it is again. We concluded that the precautionary principle works best actually in a double role of a safeguard and a compass. And actually has working also the precautionary principle operating as a source for surprises. As a safeguard and legal principle, it can justify early policy regulatory action. And as a compass and a policy principle in research and innovation, it can trigger debates upstream and research about potential impact of emerging technologies and related innovation pathways, and can lead to innovation developments and stimulate responsible innovation. And I would recommend you, for those interested, to read that. It's still available on this website that I have indicated on the slide. If we then turn to the existing regulatory regimes for GMOs, we may observe that the current regulatory regimes have set up an ex-ante authorization mechanism and an ex-post monitoring mechanism. So ex ante with the authorization procedures, and so the environmental risk assessment, and for food and feed, there is a food and feed safety assessment required prior to the deliberate release or the placing on the market. For GMOs, member states can invoke a safeguard clause should a GMO after the placing on the market appear to constitute a risk to human health and or the environment. For GM food and feed, there is actually an emergency clause that is laid down in the general food law. A long fought for opt-out possibility for member states to reduce the territorial scope of an authorized GMO for cultivation is also laid down in the GMO directive. Authorizations obtained are valid for 10 years with a possibility for renewals along the lines of the prior authorization procedure. Now, against this background, I would like to look at the NGT proposal. First question that arises is, is this proposal in accordance with the precautionary principle? I mean, that is the way that the Commission is proposed. I would like to put a question mark behind that, and that's for discussion. Second, some general remarks about the proposal itself and how it is drafted, in fact. The proposal is rather complex, so this is from a lawyer's perspective, and there can be a concern about the uniformity of the whole legislative framework on GMOs. The proposal is a lex specialis, but not always are the references to the other acts clear. The proposal replaces the procedures in the GMO legislation for NGT1 category, but not for the NGT2 category, where derogations then are made from the other GMO acts, and therefore requires much more careful reading and adds on to complexity. Also, we have discovered in discussions already with my colleagues the relations to the novel food regulation is not clear. See, for example, the recital 22 of the proposal. I'm not going into detail here. Third, there is actually, and that was also already said before by Mr. Hausling, in fact, it is not clear on the basis of what the distinction between the two categories are made. How to define the distinction between category 1 and 2 plants in a precise way. When you look at the Annex 1, it tells us that the NGT1 category sees to no more than 20 modifications. Well, Jeroen also explained that that may be difficult and that there's other cases where there's less modifications where there have been serious problems. So the question is also, what Mr. Hausling mentioned also, how this is established and by whom this is established. Looking further into more detail of what the proposal says about NG1 category plants and products, we may observe there is no risk assessment and management and no monitoring. There is only a technical verification procedure. So in my view, to answer your question, Christophe, in my view, from the reading at least, EFSA will not in fact get into a kind of a detailed analysis of any kind of risk. It is just a technical exercise. So the verification seems valid for an unlimited period, and here there seems also to be a lack of learning. There is in fact the adaptability of the rules on category 1 and 2 products. There seems to be that there is no reversibility of decisions in this category. I would also like to signal that there can be concern on the traceability, as already have been said. It is expressly stipulated that GMO and GMO food and feed legislation are not applicable. Where an NGT1 product is a food, the general food law applies. However, I still have a problem with the relation with the novel food regulation. So also for NGT1 plants, there does not seem to be a safeguard clause applicable. For category NGT2 plants, and products, the current legislative GMO acts are applicable, with, however, derogations that allow for lower requirements. For me, I must say it's a bit difficult to assess as a lawyer to what extent, so to how low these requirements then are. But it is in fact and another point in the very sorry for my voice. There is an important difference with the current regime is that the authorization for plants is valid up to 10 years, and after the first renewal, there is an unlimited period, unless, in fact, there could be a problem raised by a member state or the Commission. Important is to signal that with the application of the opt-out procedure of Article 26b of the GMO Directive is excluded for application for NGT2 plans for cultivation. So member states cannot have the territorial reduction of the cultivation. I would like to raise a few questions here. Are the sustainability incentives effective enough? Do they have a negative effect on the scope of risk assessment? Is the exclusion of hazard-resistant plants justified? Moreover, what are the effects of the proposal on international trade? Another point concerns the flexibility and future-proofness of this proposal. As we see, well, and I think we all recognize that maybe that biotechnology has seen remarkable developments within the past years. Well, is this then the proposal a fit also for future developments? Does that cover all the future developments of gene editing techniques? I cannot judge that. So,
your conclusion?
The conclusion, in fact, I would like to get the conclusion. So let us then ask us: do lessons have lessons been learned in this proposal? And I think the various crises have taught us the importance of the safeguarding public confidence and trust in the regulation of products and technology. The legitimacy of the regulatory framework is key. We have learned that actually authorization procedures based on the principle of risk analysis and the precautionary principle and monitoring plans not only serve to prevent harmful, excessively risky products and technologies from entering to the market, but also serve to generate consumer confidence in those technologies and that are under authorization procedures allowed on that market. They thus have gained, these authorization procedures have gained an important role in the building of trust in regulatory frameworks and the social acceptance of technology innovations. We should therefore bear in mind when discussing new regulatory provisions on new GMOs that we're faced not only with the question of reducing regulatory burdens, as it seems here, but also of ensuring social acceptance, societal acceptance also of new GMOs. And that was also repeated hinted at by Jeroen van der Sluis. So this, we have this recipes project that also gives guidance of how, in fact, also to include a participatory element in the application of the precautionary principle. Thank you.
Merci. Je pense qu'en écoutant les deux précédents intervenants, vous mesurez la difficulté du travail pour nous, parlementaires, dans des délais très brefs, puisque ce qui est en débat n'est pas seulement les modalités, le curseur, mais ce qui est en débat, c'est des principes même de cette législation. On y reviendra avec la représentante de la Commission. Mais je vais maintenant, on vient d'évoquer les questions d'acceptabilité sociale. Donner la parole d'abord à Olivier Andro. Donc vous représentez UFC Que Choisir, donc association française de consommateurs bien connue, qui est aussi très impliquée dans le BUC au niveau européen. Et je suis très heureux que vous ayez accepté d'être là pour nous dire votre réflexion, mais peut-être aussi pour témoigner de ce qu'est la manière dont les associations de consommateurs sont en mesure ou pas de s'approprier ce sujet, puisqu'on sait qu'en Europe, il y a eu beaucoup de débats sur les OGM il y a plus de 20 ans, que là, le débat revient de manière très rapide. Et moi, ce que j'observe dans beaucoup de réseaux, c'est que c'est un peu la course à reprendre en main le sujet pour arriver à comprendre des enjeux très techniques et pour arriver à construire des positions politiques. Je pense que c'est vrai chez les associations de consommateurs, c'est vrai aussi dans le monde agricole, paradoxalement. Vous avez la parole.
Thank you for inviting me. As you said, UFC Que Choisir is the first consumer organization in France and counts about 135 local organizations throughout France. Now, what is the position of UFC Que Choisir on the new genomic techniques? Well, I will use quite an old official position that was developed by UFC Que Choisir decades ago, when there was already a first debate on classical GMOs. And it must be noted that at that time, and still now, my organization was not and still would not be against genetic techniques and would not be against the use of GMOs, provided a certain number of important conditions. And these necessary conditions were the following and are still valid, because the first information that I gather from what was said by the scientists this morning is that the new genetic techniques are GMOs indeed. Hence, I mean, the old position of UFC Que Choisir is still valid for me. I still have a mandate to verify if the proposal that has been drafted by the Commission is in conformity with those principles. Then the first principle, the first necessary condition is that there shouldn't be any short-term or long-term risks, be it environmental risks or sanitary risks. The second condition is that the new genomic techniques should bring a global benefit for all users throughout the chain. And in particular, it should bring a benefit to the final consumer. And the third condition is that it should be very difficult or impossible to obtain a similar benefit with conventional method. Fourth condition, for producers should have the ability to choose to use or not to use such genomic techniques or such products, such NGTs. Fifth condition, in order to allow producers to be still enabled to choose to use them or not to use them, there should be strict traceability throughout the whole chain, and hence there should be an information throughout the chain from the first step till the last step of the whole chain. And the last condition, very important condition for us, is that consumers, the end consumer, should be informed of the possible presence of NGTs and GMOs in the product that is going to buy. Because, I mean, choice for consumer, information and choice is paramount for us. We demand that consumers are in the position to have a free and informed choice to buy or not to buy such a product containing or being made with the use of NGTs. So in relation with those 6 principles, what is the judgment, the assessment of the previous legislation and of the classical GMOs that did UFC Que Choisir made at that time? Concerning, first of all, the regulatory framework on GMOs. Well, we see, first of all, concerning the risk assessment, that mainly a short-term evaluation is in the present GMO regulation, and on a very limited scope. And we think that the general benefit for the whole chain, and in particular the benefit for consumers, should be taken into account, which is not the case. we think that long-term monitoring of the possible environmental risks should be taken into account, which is not the case. And we think also that the evaluation of GMOs should take into account the real-world use of such GMOs. Additionally, concerning the information for producers, I mean, one positive thing is that with the previous regulatory framework on GMOs, there is an obligation of giving the information from step N to step N+1, which is a good thing. Concerning labeling for the end consumers, I mean, we do have a labeling for food that would contain GMOs, but we have a regret because there is no labeling for feed used for animal-based products. And now concerning the GMOs that have been developed so far, I want to remind you of the promises that were made 20 or 30 years ago. And we heard that those GMOs would help get rid of hunger and famine. We would have better nutritional quality. We would use less pesticides. And what is the reality of those developments? Well, we found mainly herbicide-tolerant and insect-resistant GMOs that triggered the use of a lot more pesticides and the development of herbicide-resistant weeds. And additionally, we found no benefit for the final consumer, which is why, I mean, there is a huge majority of European and French consumers that are still against GMOs. So in conclusion, the practical position of UFC Que Choisir on the classical GMOs was and still is that they bring no benefit. It's still difficult to make the choice. So, and we are against them, against those old classical GMOs. And now, what is our position on the NGTs and on the proposal that we're given? From what we gathered this morning from the scientists that have spoken to us, we learned that the level of scientific knowledge is very basic, it's very primitive, and with huge blind spots. And now we have a major difference with the new proposal that has been prepared by the Commission. There is this distinction between Category 1 and Category 2 NGTs. And what do many scientists tell us? That this distinction is arbitrary. It's not scientific, and there is no link with the level of risk. Additionally, you cannot assume a priori the impact of a genomic modification without a full assessment. So, I mean, and it's even more, a preoccupation for us that we happen to learn that most NGTs would fall within Category 1, for which the level of requirement, as you said, would be extremely low. So the general assessment of the proposal that we make is that it's a deregulation proposal, which is in total contradiction with the precautionary principle. There is no safety assessment, a simple verification procedure. I mean, there was a statement of the EU Commission that said that there shouldn't be any safeguard measures from the member states. And hence, it means that member states wouldn't have any possibility to oppose the cultivation of such crops on their territory. Concerning traceability, for Category 1 NGTs, there would be no specific traceability and no specific information given from one state to the next, which means that it would induce a huge risk of contamination for conventional and organic products, and it would induce a de facto general contamination. So, concerning the choice for producers, well, there is no choice that is given, and who will pay if organic producers get contaminations in their crops? We don't have any answer to this question for the moment. And concerning consumers, there shall be no labeling, no specific labeling in the case of category 1 entities. So, hence, there will be no possibility for consumers to be informed, and then no possibility to make an informed choice to buy or not to buy products containing such entities. And I want to remind you that this is the main demand of consumers, and hence the main demand of UFC Que Choisir. I mean, to give consumers the choice and to have an informed choice. Concerning the benefit for consumers, we didn't see why the promises that were made years ago would be very different from those those who advocate for the development of NGTs. We don't see why they should be different. I mean, we hear nutrition, safety aspect, price hike. Nutrition, it's a problem of less sugar, less saturated fat, less salt. So it's a problem of industrial recipes and unhealthy marketing, mainly to children. Safety aspect, it's a problem of hygiene. So it's a problem of official monitoring. It's a problem also of food additives, like nitrates. So it's a problem of legal restriction of use. It's also a problem of components of food packaging, like bisphenol. So it's still a problem of regulation. And concerning price hike, well, it's mainly due to the war in Ukraine. So it's a problem of also of autonomy and EU production, national production and local production. But all these problems do not have any relation with NGTs. So what we think is that so far UFC Que Choisir is not in favor of those NGMOs and demand that all NGTs go through a full authorization procedure. We want member states to be allowed to pause in such NGTs, and we want a full traceability and an on-pack labeling for all NGTs, category 1 and category 2. Thank you.
Merci pour cette position extrêmement claire. Eric Gall représente ici IFOAM, donc l'Organisation européenne des producteurs de l'agriculture biologique, et il va évoquer l'avis de ces producteurs. Éventuellement, peut-être aussi avoir une petite extension sur les autres filières non OGM, signe de qualité. Le bio est aussi un signe de qualité, puisque le bio est un secteur emblématique, mais n'est pas le seul à se poser la question de sa protection vis-à-vis de ce type de produit. Vous avez la parole.
Indeed, and I will also speak in English. Thank you very much for your invitation. Indeed, we represent around 200 members across Europe, not only organic farmers organization, but also processors, retailers, and part of a research community involved in developing organic farming. Organic agriculture nowadays is around 9 to 10% of European agriculture. And that represents between 10 and 15% of farm employment across Europe as well. And last year it was a market of 56 billion euros in Europe. My point is that we are not talking about a sect of hippies. We are really talking about farmers and companies who are innovating and who are experimenting methods of food production that do not have a negative impact on nature and on the health of people. And we also have a target of 25% organic farming proposed by the Farm to Fork strategy in the European Union. There is a reason for that. The reason is that the benefits of organic productions are clearly established by the scientific literature. These were the conclusion of an inquiry by the French Court of Auditors that was published last year. Benefits for biodiversity, benefits for water quality, benefits for soil health, and so on. And this is in contradiction to the alleged benefits of NGTs and GMOs. Organic production has clearly established benefits. And this is also why, for example, water agencies in France are funding the conversion of farmers to organic in water catchment areas in France. I'm saying that because I want to clarify two things, first of all. Our members are really worried and quite shocked by the calendar that was communicating in the last days to discuss about this proposal on NGTs. I think with all the interventions we've heard this morning, it's clear that this proposal could have very important implications on many fronts. And I mean, we are still living in democratic societies where we attempt to have rational debates about the consequences of these issues. And I'm sorry, but having to figure out with such tight deadlines whether we should deregulate some of these genetic engineering techniques is not responsible policy making. So we are really appealing to policymakers to take the proper time to assess the consequences of what they will decide on this front. And I must say that I was reading the draft report of Ms. Pulford, the rapporteur from the EPP group on that. And we were also shocked by two elements. I mean, first of all, by the proposal that any reference to genetic modifications will be deleted from this proposal on NGTs. You mentioned that the legal framework was about, also ensuring public trust and public acceptance. We really get the impression that this proposal is about manufacturing ignorance, actually. Because if you take away the right of citizens and consumers to know what's in their food, if you take away labeling and traceability, then people don't have a choice, and they don't know what's in their food as well. And we know very well that that's one of the objectives of a biotech industry. When you talk to industry people, they don't have any problem with traceability. They are confident they can ensure traceability all along the production chain. They just don't want it because they don't want consumer labeling, because they consider that labeling NGT products on food products is a way to stigmatize these products. The second point I would like to clarify, also on the basis of what I've read of Ms. Pulford's proposal, is that it's very important that there is a clear ban of genetic engineering and of NGTs in organic production. And this is clearly one of the very positive points of the Commission's proposal. I've said it before, and I will say it again. The Commission's proposal acknowledges that producers who do not wish to use NGTs should be allowed to do so. And for this, you need to have a minimum of mandatory information about the presence of NGTs in the breeding and on seeds. That's not enough, very clearly, and we clearly ask for the traceability to be extended to all actors in the production chain, not only to stop at the seed level. But we are quite shocked by the proposal from the EPP rapporteur to get rid of this ban in organic production and to claim, like it is written in a proposal, that this ban of the use of genetic engineering in organic is not science-based. This is what is written in Ms. Pulford's report. I find it quite appropriate that you started this panel by reminding about the importance of a precautionary principle, because organic farming is based on 4 principles: ecology, health, fairness, and care. And the principle of care is basically about implementing the precautionary principle. This is why the use of synthetic pesticides is banned from organic production. How many synthetic pesticides were assessed by scientific authorities like EFSA and were deemed safe and authorized to be put on the market? And then only 10, 20 years later, there were enough publications accumulated to show that they were actually risked for biodiversity, for the health of people, and then they were banned. Just think about neonicotinoids. So would you have said 30 years ago that organic agriculture was not science-based because it was refusing to use neonicotinoids even though they were authorized at EU level? Clearly, no. And this is the reason why organic producers, and many producers who are not organic across Europe do not wish to use genetic engineering in their production process as well. So they are having a precautionary approach is clearly more science-based than proposing to deregulate NGTs on the basis of whether there are less than 20 modifications, for example, as well. So we really urge policymakers not only to maintain this ban in organic production, but precisely to provide all farmers in Europe, whether they are organic or not, with the legal and technical means to choose not to use NGTs if they don't want to do so, and if this is what their consumers demand from them as well. And for this, we need to have a traceability system all along the production chain. We need to have consumer labeling as well. And on top of that, just let me finish on that as well, because I hear some MEPs saying, I have organic farmers in my constituency, and they're asking me, why are they not allowed to use NGTs? And allegedly, these organic farmers are demanding politicians to use NGTs. I'm sure, there are such organic farmers, and we heard some of them in the European Parliament as well. But in the organic community, we had many discussions about that. And we even had a resolution adopted last June where we had a debate and a vote. And 97% of our members voted clearly in favor of not allowing NGTs in organic production. So once again, we live in a democracy, and clearly you can always find individual farmers and organic producers who believe that NGTs are interesting for their production, fine. But the vast majority of organic producers do not wish to use genetic engineering in their production process. And it's even more important at a moment where the organic market is impacted by inflation and is not doing very well. It's even more important to protect the trust of consumers in organic production and the reputation of organic products as well. So let me say one thing. If an organic farmer wants to use NGTs, this farmer can stop being an organic farmer. Nobody forces anybody to be an organic farmer and to follow all the organic principles and all the detailed regulations and provisions that an organic producer has to follow. And conversely, nobody should force an organic farmer, or any farmer for that matter, to use NGTs if they don't want to do so. But this is what the proposal would amount to if there was no traceability and no information about the presence of NGTs in seed in particular as well. So that's why it's really crucial to maintain this ban of NGTs in organic production and to offer all producers, once again, because it's not only about organic farmer, like you said, Mr. Clergeau, the means not to use NGTs through information and through traceability. And my last point will be about coexistence measures, because traceability and information of consumers and producers is fundamental. But if we have learned something from this legal framework on GMOs in the last 20 years, is that there is a need for measures to protect farmers who do not wish to use NGTs from contamination and the economic consequences linked to it. We had a legislative guerrilla for 10 years in the European Union because in 2001, the Commission refused to legislate on coexistence, and member states were using safeguard clause to ban nonetheless the cultivation of GMOs on their territory to protect organic production, to protect conventional production as well. And that's why in 2009, the Commission came up with this proposal to give the possibility of opting out from cultivation as well. If we don't want to be in such a scenario again, we really urge policymakers and members of the Parliament to provide a clear legal basis for member states to adopt coexistence measures also for NGTs of all categories. Thank you very much.
Merci beaucoup. Donc, je vais donner la parole à Madame Sanchez pour avoir les positions et surtout les réactions de la Commission à ce que nous entendons. Puis ensuite, on ouvrira le débat. Évidemment, beaucoup de sujets ont été évoqués, quasiment tous les sujets de la législation. Moi, je voudrais juste en rappeler quelques-uns. D'abord, la question des bénéfices. Pourquoi je dis ça? Parce qu'il y a un position paper du groupe Renew, qui est connu depuis de nombreux mois, où la position initiale du groupe Renew, c'était de réserver l'autorisation des nouveaux OGM à des nouveaux OGM qui auraient un bénéfice contribuant à la lutte contre le changement climatique. Et dans leur vision, c'était y compris catégorie 1. Et cette vision n'a pas été retenue, puisque la Commission, dans ses exposés, considère qu'il y a une possibilité de bénéfice global, mais il n'y a pas à caractériser un bénéfice dans la catégorie 1 pour pouvoir déposer un dossier. Ça, c'est ma première question. La deuxième question porte sur la prise en compte de l'état de la science. Et ce qui vient d'être rappelé par les uns et par les autres est très important. Il y a des évaluations scientifiques faites par l'EFSA. La Commission s'est basée là-dessus, mais avec des modalités de décision, c'était rappelé par Hélène Vos, renouvellement illimité, absence de clause de sauvegarde vis-à-vis des États, qui rendent d'une certaine manière le dispositif irréversible et incapable de prendre en compte l'avancée de l'évaluation scientifique, alors qu'on est en situation de controverse scientifique. Et je ne sais pas si ceux qui sont intervenus aujourd'hui avaient la vérité, mais la controverse existe. Donc comment est-ce qu'on prend en compte cette question de la controverse, de l'instabilité de l'état de la science pour la définition de la gestion du risque, puisque ce n'est pas le cas actuellement. Troisième élément, mais je n'en ai pas beaucoup d'autres, catégorie 1, catégorie 2, c'est extrêmement contesté. Et du coup, Thèse Biotech a proposé une autre approche de la définition de catégorie 1, de catégorie 2, qui est d'établir par des méthodes scientifiques que la plante est équivalente, plutôt que de chercher à définir des critères probablement par définition arbitraires. Donc ça, c'est une contre-proposition forte. Je ne sais pas du tout ce que vous en pensez, puisque c'est la première fois, en tout cas pour moi, qu'elle vient dans le débat sous cette forme-là aujourd'hui. Et je termine par le bio et l'acceptabilité sociale. Monsieur Gall vient de rappeler que c'est 25% de l'agriculture européenne dans les objectifs politiques de la Commission. Et nous savons tous, et vous y compris, que s'il n'y a pas de traçabilité et mesure de coexistence, l'interdiction dans le bio n'a pas de sens. Donc c'est très bien que la Commission ait introduit, un peu à la dernière minute et contre beaucoup d'avis, cette interdiction des NGT dans le bio, mais est-ce qu'on n'est pas, y compris d'un point de vue juridique, à mi-chemin et dans l'obligation soit de revenir en arrière, ce que propose la rapporteure PPE, soit d'aller jusqu'au bout avec la traçabilité et les mesures de coexistence. Et puis dernier élément, c'est est-ce qu'on peut encore aujourd'hui faire une législation pour rendre plus facile la mise sur le marché d'une technologie? Vous voyez, je fais l'effort de ne pas utiliser le mot dérégulation, alors qu'il y a d'énormes enjeux d'acceptabilité sociale aujourd'hui. C'est-à-dire qu'on risque d'aller vers une conflictualité forte autour de ce sujet-là si la législation reste en l'état et si on ne réintègre pas toute une série d'éléments de régulation tels qu'ils viennent d'être appelés par les uns et par les autres. Est-ce que c'est possible pour la Commission d'aller au bout d'un processus face à un tel niveau de contestation de part importante de la société civile et qui mettent en lumière des choses différentes de celles qui sont apparues dans votre consultation et dans vos évaluations préalables? Merci.
Thank you very much. I have 2 hours.
No.
Okay, I'm going to try, but there has been indeed an enormous amount of issues raised, and I would really love to go back to quite a few of them. Before I start, however, thank you very much for the invitation and for the opportunity to discuss with all of you today. I'm going to try to be structured and fast. Let me first start with a very general issue in reaction to a couple of things that have been raised. We come with a proposal that, in our view, is very solidly underpinned both by evidence and by wide consultation. There has been some statements about bias, over-representation of certain stakeholders. I mean, I know many of you in this room. We have talked to all of you. We have exchanged letters, etc. This is a 4-year process now, or even more, with the Commission study, and I think all the voices have been heard. It is a difficult debate where there are different positions, that's for sure. The issue of evidence, this is a crucial issue, and it goes directly into the discussions of this panel on the precautionary principle, because, I mean, for starters, the principle is about science-based policy and about how to handle when there is uncertainty about risks. I think at the very beginning of the event, Astrid opened saying we need to take into account all of science and not just the majority opinion. We agree with that, and I think Ana Afonso from EFSA has been very clear to say, for example, on the issue of unintended effects, that all of the evidence out there has been considered. I think a lot of the debate that we are having today in this panel is about this issue. We are following very much, certainly the advice of EFSA, because they are the scientific agency in this field in Europe. And EFSA has worked on the basis, always in their opinions, of public consultations, of responding to comments. And the conclusions of EFSA, and I would like to come back to one of your questions earlier. Anna said before, there are no new hazards or specific hazards of these techniques, which is a very fundamental underpinning of the proposal, in relation to conventional breeding or established techniques. And you were asking, what does that mean? Our proposal acknowledges, and we think that's solidly underpinned by EFSA's work, that new genomic techniques can be used in very different ways, and in some instances they will lead to products that could have been obtained possibly in a much longer time frame through conventional breeding, and in other cases they will lead to products that cannot be obtained with conventional breeding and that are much more similar in terms of the risk profile to the current transgenics. This takes me now into the criteria, and I've heard a lot of comments about the criteria being unscientific. What we have tried with the criteria is to identify what are the type of modifications that are documented to be achievable with conventional breeding. Taking this statement of EFSA that there are no new hazards compared to conventional breeding or established techniques, we exclude, for example, from category 1 completely intragenic plants, because EFSA has considered that those are closer in terms of the risk profile to transgenic. We also exclude from category 1 random cisgenesis. I think the type of modifications that we've included in the criteria, I have not heard the type of modifications themselves questioned. What we hear questioned are the thresholds, why 20, why not 21 or 19? I mean, obviously 20 is a choice, and when you are between 19 and 21, whenever you make a choice, whether it's the age of voting or anything else, you can fall the day before or the day after, right? But we have made a choice which we understand is a conservative choice. The literature shows that the number of mutations and the size that can be obtained with conventional breeding is higher, but we are taking into account also the possibility of obtaining, what would be the likelihood of obtaining that number in that combination. Our paper on the literature we have analyzed is out. I have to say that a lot of the reactions that we are getting from the scientific community are a little bit different from what we have heard today. We are rather being told that the threshold is too low. So, for example, I think yesterday or the day before there was the statement of the German Independent Expert Committee on Biosafety, the SEIKBS, and their main comment is it's too low. So we have made a choice, and we have tried to make a conservative choice. And that is already part of how the precautionary principle is applied in this proposal. On the precautionary principle, I want to raise two elements which are important dimensions in the application of the precautionary principle as construed by the court, and which have not been mentioned, or at least I haven't caught them in the discussions today. Proportionality and non-discrimination. The precautionary principle is a guide, and I like very much the use of compass by Ellen earlier. Is a compass in situations when we want to avoid harm if there is any uncertainty on risk, but it's not a principle that says you have always need to take the strictest possible measure. Proportionality and non-discrimination are part of that, and they are very much part of the approach of our proposal. This is why we go with two categories. This is also why we try to create predictability with this criteria. I mean, you were asking me about the counter-proposal from Test Biotech to do it case by case. I mean, what I would say in response to that is, I mean, we still need some criteria in the legislation. I don't think that in an area like this, the legislator would say, okay, we don't have any criteria in the legislation, and case by case, let's decide. we have tried to be much more specific with our criteria in our legislation than other third countries that have high-level criteria of equivalence to conventional breeding, for predictability reasons, but also as part of how we reply to the precautionary principle. What we are trying to do is something which is proportionate to the different types of products that can be obtained with these techniques. We fully agree with what you were saying, Mr. Clergeau, at the beginning. This is not about the techniques, yes or no. It's what are the products being obtained. And the issue of non-discrimination comes in as well. Some of the discussions, do these techniques have unintended effects? That's not the question. The question is, do they have unintended effects that differ from those of conventional breeding? And in those cases where they don't, it would be discriminatory to treat the products of new genomic techniques in a different way from the products of conventional breeding. That's a little bit how we see the issue of the precautionary principle. I also want to say, because in the two presentations on the precautionary principle, a lot of issues were raised, and some issues, in my mind, are not related to the precautionary principle. The opt-out in the current legislation is not precautionary principle. It's not about safety, and the legislation is very clear about that. These are products that EFSA has considered to be safe. But let me go now to consumers and to traceability, organics, etc. I think we fully agree that guiding principles here relate to the absence of risk for consumers and also benefits for the final consumer. I also think that we need an open debate with the public about these technologies, and that's something that we should all engage in and promote. I don't think myself that today there is a European citizen view on NGTs. I think there's mostly lack of awareness. There was very interesting social research done by EFSA on this that I don't have the time to go into, but you can look up if you wish. But I would also like to bring another dimension into this discussion that I have found a little bit absent in the discussion today, which is the issue of what tools are we giving farmers to cope with current challenges? I mean, consumers and benefits for consumers are also about increasing consumers' choice of safe and sustainable products. It is also about giving farmers the tools to continue feeding us and giving us choice. And I think that's a very important part of this debate as well. We have gone for a system, and of course we've made our choices, and the co-legislator will make the ultimate choices because we just propose. We have made a choice to label as GMOs the category 2 products which are subject to a risk assessment and an authorization. But we have considered that the labeling of category 1 as GMO products is not appropriate. The GMO label already carries a meaning with our consumers. It's linked with products transgenics, which is what has been authorized so far, which have little to do with the kind of products that will fall in category 1. And also we need to take into account that if we are going to inform consumers about something, and of course, I think freedom of choice for consumer and information is key, but we make decisions all the time about what information goes in the label. We don't inform about every manipulation or every way that our food has been produced. If we put a label in a product, we need to be sure that that label is accurate. And here is where we enter also into the issue of detection and traceability of these products. We have a clear view from the European Union Reference Laboratory and the network of laboratories that there are challenges in this regard. And by the way, I would like to say in response to the previous panel, to Sarah, I don't see a contradiction between acknowledging the challenges today and putting Commission funding into detection. We were criticized in previous Horizon programs because we didn't have funding for detection. Now we have funding for detection, and we are being criticized because I'm not sure. But anyway, today, what we are told by the European network of laboratories is the major problem with analytical technologies is that they cannot discriminate products derived from plants with identical sequence modifications obtained by different techniques: targeted mutagenesis, cisgenesis, conventional breeding, natural mutation. If we are committed to science-based policy, we need to acknowledge this. So a full system of traceability of the category 1 products that are the most likely to occur in nature, it would require full segregation of supply chains. There would not be an analytical conclusive method. It would not be possible for the organic sector to conclusively demonstrate lack of presence. I think this would create a burden which is not justified by safety considerations, that is disproportionate to the aims and to the nature of category 1 products. And I realize you've been very patient with me, so I'm going to stop because I've exceeded my time already. Thank you.
Non, merci beaucoup. Pour moi, c'était très important que vous ayez le temps de répondre aux points essentiels. On va prendre quelques questions de la salle. Normalement, on devait s'arrêter à midi, mais la salle est réservée à 12h30. Donc, si les traducteurs, que je remercie, s'arrêtent à midi comme c'est prévu, on pourra toujours prolonger de 5-10 minutes en anglais. Je vais faire juste une remarque avant de donner la parole. Il y a un débat sur les méthodes. Information des consommateurs, ce n'est pas forcément label en gros sur l'étiquette. Il y a plusieurs manières d'informer les consommateurs et je pense qu'on est tous prêts à en discuter. Et ça a été dit, il y a une alternative à la détection sur les produits qui est la documentation complète sur la chaîne de valeur. Elle pose d'autres problèmes, mais cette alternative existe. Donc ça, je pense, j'entends votre réponse, mais elle ne ferme pas la discussion sur ces chemins possibles.